ஹலோ சி சரி சார் ஓகே ஜிகே ஹலோ கேட்குதா சார் எது புரியுது ஜி எதுக்கு கெஸ்ட்டு வராங்களா சொன்னாங்களா கெஸ்ட்டிலே யாருங்க அப்படியே இங்கே வந்துடும் மேடம் கெஸ்ட் நேராக இங்கே வந்துடுவாங்களா வெளியே ஆர்டர் பண்ணுறேன் எதுவுமே இல்லை தானே கெஸ்ட்லாம் இங்கே அப்போ டேரெக்டாக நீ வெளியிலே போக வேண்டாம் சரியா நேற்று காலையில் இப்போ வந்து நீங்களா அந்த லிங்க் வந்து நீங்கள் ஒரு உங்கள் ஃபோன் நம்பர் பார்த்தீங்களா உங்கள் ப்ளீஸ் அதுக்குள்ளே போய்ட்டு நீங்கள் பேசுங்க அந்த லிங்கை வந்து இதில் ஷேர் பண்ணி விட்றேன் பின்னாடி ஸ்க்ரீன் இறக்கி விட்டு ஓகேங்களா ஆமாம் எனக்கு என் மெயிலுக்கு ஷேர் பண்ணிங்கன்னா நான் லாகின் பண்ணி ஓப்பன் பண்ணிவிட்றேன் இங்கேருந்து நான் பண்ண முடியாதா இங்கேருந்து பண்ண பண்ணிக்கலாம் ஆனால் மூஞ்சி உங்கள் திருப்பி வச்சுக்கிறேன் ஆனால் இங்கே பண்ணாதீங்க இங்கே வந்து லைவ் போய்ட்டுருக்கு 2 காரணம் ஓகேங்களா லைவ் யூடியூப் லைவ் போய்ட்டுருக்கு ஒரு மிஸ்டேக் லாகின் ஆகிடுச்சுன்னு வேறு மெயில் கனெக்ட் பண்ணி வச்சுக்கிறாங்க இதில் என் மெயில் கனெக்ட் பண்ண முடியாது என்ன பண்ணிட்டோம் நீங்கள் வந்து இன்னொரு இடத்துக்கு உட்காந்து யூஸ் பண்ணணும் நீங்கள் வந்து அந்த லிங்க்கை மட்டும் இதில் பேசணும்னா பேசலாம் உங்கள் மெயில் போட்டு உங்கள் மெயிலுக்கு கனெக்ட் பண்ணிடுங்க உங்கள் மெயிலே கனெக்ட் பண்ணிடுங்க உங்கள் மெயிலே கனெக்ட் பண்ணி இதில் யூஸ் பண்ணிங்கன்னா அந்த டிஸ்பிளே அது என்ன சொல்லணும்னா இன்னொரு வந்து க்ரோம் தான் யூஸ் பண்ணணும் க்ரோம் மீதி எல்லாம் இன்ட்டுமா கொடுத்துறாங்க லைவ் போட்டுருக்குது க்ரோம் மட்டும் யூஸ் பண்ணுங்க தெரியாமல் இங்கெல்லாம் வந்து இங்கே போகிறதா இல்லை போயிட்டு லைவ் போயிட்டு ஒன்லி க்ரோம் தான் ஆனால் திரும்பி எதுவும் மிஸ்டேக் பண்ணிக்க என்ன பண்ணுறாங்க வந்து டிப் கொடுத்துட்றீங்க அதுதான் ஹலோ சி சிகல் ப்ளேவா எதுங்க அது போட்டு வராங்க அதுதான் போட்டு வரோம் அது பண்ணிக்கலாம் கரண்ட் இல்லை வரும் ஹலோ ஹலோ சி Audio. Hello, hello. Hello, hello, hello. Chick, chick, hello. Hello, chick.
I request everybody to assemble in the conference hall. Very shortly we are going to start our today's session. Please be seated. Good morning. Sir from PhD College and Dr. K. Murugan sir from MK University and Dr. Hema Aishwarya Madam, Chennai, scientist. And today also we are going to have a start our next technical session today by our honorable, eminent, invited speaker, Dr. A. Murugan, Professor and Head from Department of Microbiology, Periyar University, Salem. First of all, I would like to take this wonderful opportunity to introduce Dr. Murugan sir. Sir has acquired BSc Zoology from Madurai Kamarajar University, MSc Microbiology from Manonmaniam Sundarnar University, Tirunelveli, PhD in Microbiology from Manonmaniam Sundarnar University, Tirunelveli. Sir has been cleared set qualified from Bharadasan University, Tiruchirappalli. His area of interest are therapeutic compounds from gut microbes, metabolics. Sir had undertaken as a principal investigator for so many major and minor projects sanctioned by UGC, DBT, DST, and he had completed so many major projects also. Sir had published more than 20 papers in national and international journals with high impact factor. Book chapter authorized is 8 numbers. Until now, Sir had submitted 28 microorganisms in gene sequencing and he acquired the accession number also. Sir had produced 6 PhD scholars under his able guidance. Ongoing PhD scholars is 5 numbers. MPhil produced scholars is 18. To his credit, career achievements, he organized many national and international seminars, conferences, workshops, awareness programs, faculty development programs in national and international level. As well as he acted as a research program for many training programs and workshops such as a few topics such as mushroom cultivation, advanced techniques in biotechnology and goes on. And such as global warming, quorum sensing, biofuel production, etc. Sir has held so many professional positions such as he is a director in Periyar University of Distance Education, convener of Academic Quality Assurance Committee, as well as Inspection Committee of Industry Collaboration Progress in Periyar University. He is a director of Center for Survey of Vivekananda Studies, and he is a BOS member in PG Board of Studies in Periyar University, Sri Venkateshwara University, and as well as Sir is a board of subject expert member in our institution also, in Selvam College, Padmavati colleges, and as well as so many autonomous colleges in and around Periyar University. Sir had attended UGC sponsored workshops, short courses, and as well as he had acted as a many resource person, and he had visited as many countries like Singapore, Malaysia, China as a resource person as well as a visiting professor. Thank you. Dr. A. Murugan sir is an excellent convergent, debonair personality. Today this decorum personality is before us to deliver a lecture on empowering institutions signaling pathway through short chain fatty acids. The glaring countenance of our participants clearly reveal to hear your wonderful lecture, sir. Let us invite him to spark the gatherings. Please give a round of applause to spark the gatherings.
So,
Before that, I invite Dr. Anbagan sir and Dr. E.G. Wesley sir to honor our resource person Dr. Murugan sir with a shawl and shield. Welcome, sir.
at the outset, I feel very happy to be here. Since my earlier career were here, started from MGR Art and Science, then I was pulled by a great gentleman to this Department of Biotechnology. I believe it was 2003. We were all working together, Dr. Wesley and Dr. Anblagan and many other colleagues. They were all settled in different places. One of my junior was Dr. P. Ayyappan, I believe. He's in Malaysia. So, that period was very, memorable and green in my heart. Almost some 400 and plus students in the Department of Biotechnology alone. We will be running all the day, almost 7 hours. We don't even have a time to drink coffee or water. In between, we'll have half an hour break for lunch. That also will have some of the discipline work assigned by the management. We'll be very busy with that. I was able to stay here only for 11 months. Okay, that is my short temper, I would say. So from that, I have matured enough to move around different places, and from here and then move to Muthayammal to Selvam, from Selvam to Kamarajar University, then back to Periyar. So it doubles my happiness by the beautiful introduction. All that is given by me. She has not missed even a single word. I must thank her for such a wonderful introduction. I'm so happy that I'm here in the institution which has crossed almost 30 years of its service to the society. My talk will be on a knowledge sharing. It is not my original data, but our data are there, which are very confidential. We will be publishing it once we get approval from ICMR. Since we do research on human subjects directly, it needs a lot of permission from the ICMR. So we are waiting for such approval from ICMR itself. So to let them get released, our work focuses on the gut bacteria, which gives you longevity. Longevity in the sense it is not 100 years. A human can live more than 580 years. You cannot believe it. But such a wonderful work we have, it is possible. I'm sharing to people who are going to lead this nation. Right? Our period will be another 50 or 60 years. But again, the title of the conference is Human Pathogenesis. But my talk is purely on the diseases which human get, not by the microbes. We have lot and lot of the diseases which are, killing the human being, which are all non-communicable, which are all metabolic disorders. If you Google it, you will find diseases which kill the humans in large number. Top 10 diseases which kill the human at the global. The first one, what would be? Can anybody guess? You can tell me. உலகத்திலேயே அதிக நபர்களை கொல்லக்கூடிய காவு வாங்கக்கூடிய ஒரு நோய்னா எதுவாக இருக்கலாம் சார்? பாருங்க, ஸ்மால் பாக்ஸ்ன்னு சொல்றாரு. இட் இஸ் அ வைரல் டிசீஸ், ரைட்? அதுவா என்கிட்ட டேட்டா இருக்கு, காட்ட போறேன். நோ மோர் சீக்ரெட். யூ கேன், இஃப் யூ கூகுள் இட் யூ கெட் இட். வேற இந்த சைடு இருந்து யாராவது, what would be the disease which could even claim more and more people even now? See, cancer. That's how we miss the important information we supposed to get. We are all living with a borrowed knowledge. Next slide. We are living with a borrowed knowledge which comes through YouTube, knowledge comes from WhatsApp, knowledge comes from Facebook, which are not authenticated by anybody. Nobody checks whether the data are true or false. Fake information comes, we chaos it, we forward and make total population go into crazy after such fake information. See, here is the next slide. Here is the data. You can see it. No such disease, whatever you said here. Top 10 diseases which kill the people in large number. It is in trillion. We are going to see it. It's in trillion. So, first disease which is cardiac vascular disease, CVD, we would say, which comes by different mechanism. All that we are going to see. Next, you see, that is also related to cardiac vascular disease. Then you see a diabetes mellitus. Okay, this is another important culprit which also brings cardiovascular disease, right? So inflammation, that leads to diarrhea. Diarrhea is another important problem which we often get, which claims large number of population. So in a way, we have a data which it says non-communicable diseases claim more life than any deadly diseases what we believe, including chikungunya, COVID, dengue, okay, and all that, the world diseases, whatever we have, and also the diarrhea in the list. So communicable diseases, we have large number, but the total death occurring at the global is cardiovascular diseases, right? Next slide. It is not my data. You can Google. WHO has data, and also CDC, another website. You can find the data, okay, in a updated version. Even now, what is the disease is predominant at the global level that you can identify by visiting CDC. You see here, the cardiovascular disease is the top one. Almost 17.79 million, that is in 2017. The data are available in the website up to 2019. The current data is here to receive. Right, in a way, you just come down, you will find the malaria, right, and other diseases. The bacterial diseases are very less going down, right? Very less. But almost the diseases which we all have are all not in our idea. So we'll have to understand the disease which kill more and more are all not bacterial or viral or not communicable diseases, which are all non-communicable diseases. How we get these diseases? These diseases are become more and more prevalent than the earlier days. Earlier days, what I mean, some 5 million years ago or 4 million years ago. So what is the reason for such diseases? Very alarming, very, important information you must understand. The habit, food habit is the culprit here. The main food which we eat brings a lot of change in the metabolism. That metabolism culminates into diseases, non-communicable diseases. Right? The next slide. So people with a family history of heart diseases are not sure to have CVD. Similarly, genes are not a matter, physiology not a matter, smoking factor is not a matter, right? But then what is the matter? What we eat is a matter. Next slide. So these are all myth on it. So global burden of cardiovascular disease or cardio diseases, you will find the intake of the fat diet, the diet which rich of fat, the diet which rich of calorie with less nutrient is cause for such increasing of cardiovascular diseases. This slide, there is TMO. You will understand better. Trimethylamine N oxide. This is the chemical which is formed in our system when we have more carnitine as well as lecithin-like substances. Carnitine is rich in red meat. I'm sorry, I'm not hurting the people who are non-vegetarian, but the data is here like this. Carnitine is rich in red meat. This carnitine is metabolized, and produce a chemical called TMO. This TMO causes sclerosis. You will find here, trimethylamine N oxide causes sclerosis, meaning inflammation and break in the artery. That causes inflammation in the artery, leading to formation of clot, thrombosis, we'll say, thrombose. This thrombose leads to formation of many clot here and there. That clot leads to stroke, cardiac arrest. Right? This is the story which we did not understand for a longer period. Many people have died after eating 2-3 times of red meat. They for Diwali, like festivals, people take some 3 kg, 4 kg of red meat. They eat 3-4 times in 2 days, 3 days time, and the amount of DMO formed is proportionately high that would lead to stroke. If at all they are a hyperactive patient or hypertension cases, right, or cardiac failure already they have. This will increase the possibility. That is how the data goes. See, it is the reference. You can also find it. Next slide. So very well matches with the rest of the symptoms. This TMO causes cholesterol accumulation. So you can ask me how this cholesterol and the cardiovascular is connected. Cholesterol is an essential fatty acid which is secreted by our body. Imagine this is the building which is constructed some 10 years before. If at all there is some crack, our management people will examine and then inform to some people, Manson people will come and apply cement. Likewise, the artery which has some crack inside need to be attended by our system. A cholesterol is a cement which seal the cracks. Likewise, whenever there is a damage inside, cholesterol is being produced by our system in order to heal it, shield it. Okay, and cementing is the work. In a way, we have this similarly foam cell formation, which is also another important function to protect the damages. Again, we have a platelet hyperresponsive, then vascular endothelial inflammation. All that happens when we have TMAO in high. Okay, almost it will be a threshold. Anything it is in a threshold, it's okay to the system. If it goes beyond the threshold, it's going to be the problem. நம்ம தமிழில் சொல்லியிருக்காங்க, ரைட்? எதுவுமே அளவுக்கு மீறினால் அமிர்தமும் நஞ்சு. ரெட் மீட் சாப்பிடக்கூடாதுன்னு இல்லை, there is limit, right? So it should not accumulate TMO on a higher level. That is the thing, right? And you see here, there are bacteria which also influence this TMO formation. There are good number of bacteria, Campylobacter, Corynebacterium, and Provitella, which are all the common bacteria which enhances the formation of trimethylamine oxide. Otherwise, it will be a trimethylamine in the system. Trimethylamine does not cause cardiovascular disease. Sclerosis will not, will be the case with TM alone. Trimethylamine does not. But interestingly, when there is a change in the population of our gut, we have a gut bacteria that I'm coming to that next level, next session. So we have a gut bacteria. This gut bacteria contribute TMAO production. Otherwise, it will not be this thing. People who are taking medicine for diabetic also have more and more of TMA, trimethylamine. You can Google it. Metformin produces trimethylamine. So people who take diabetes mellitus also highly prone for CVD. You can check it. Okay, so it is, see how the evil come into the life of human. You see, we didn't know. Even the physician do not know. The people who prescribe this do not know. But data, scientific information comes in. This metformin is converted into methylamine in our system. That they have not studied. What they studied, it is going to trigger the conversion of glucose through your urinary system. That's it. Okay. What they wanted, they wanted to send the glucose out of the body through urine. For that, chemical is used. But inside it has a strong negative side effect, which is going to climb life. Next. You see, the association of gut bacteria and the progression of cardiovascular disease. We have found molecule, participation of gut microbiota. You see less and less of alpha diversity. Bacterioids. We have a less and less of bacterioids. Someone can check it. Nowadays, this next generation sequencing services are available for 3,000 rupees. You can take the blood sample of cardiovascular patient or a fecal sample, give for whole genome sequencing. 5,000 rupees to 3,000. You can check how their body microbial load. Okay, what are all the microbes present in the blood? Even blood has. Right? Similarly, the gut bacteria. What is the gut bacteria? All that you can check. So less of diversity. And Provitella, you see, just now we were seeing Provitella. And similarly, you see Streptococci, Enterobacterias in Lactobacillus, and Clostridium subcluster, you see. The good bacteria number, bacterioids, goes down. When you are prone, when you have this chemical and more, good bacteria goes less and less. That is the cause for the disease, right? As soon as you are in a family which are very peaceful and economically in a, settled well, you don't have much problem. You are in a family where a lot of chaos, a lot of trouble, a lot of mental disturbance, then you are susceptible for many other diseases. Reason we have data. Okay, I'll tell you that also. So in a way, you see, participation got metabolites, you see, less and less of short chain fatty acid. I have brought a lot of slide on it. When you have the altered bacterium, disturbed bacterium, you have less and less of the beneficial metabolites. You have more and more of a dangerous chemical in the system. Dangerous chemical is what? TMAO, trimethylamine oxide. Next slide. So very, very, disturbing data which are coming. It is not mine. It is all there in the Google. You can identify the paper, identify it. So there is imbalance in the bacterium, especially in the gut. So the more and bacteria what we have in our system is in the gut, right? So what about other organism? Other system. See, next slide. So, 1 minute. Previous slide. Previous. Okay. You see, not only the cardiovascular disease, many mental disorders like, Parkinson, Alzheimer, autism, just see, and obsessive compulsive disorder and post-traumatic stress disorder, ulcerative colitis, right? All that are caused by a altered bacterial population, not by any other chemical. If the bacteria is altered, the total things go reverse, right? It is in a opposite order. Next. So that we call as dysbiosis. Okay, dysbiosis. Altered microbial population is known as dysbiosis. Means disturbed or not in order. Biological microbiome load is not in order. Okay, so left side you see all the good bacteria. Right side you will have a bad bacteria. So when there is bad bacteria, the endothelium, okay, our endothelium meaning the intestine, which is well protected by a mucus layer, there is a bacteria called Lactobacillus mucinus, which is one which produces a mucus layer. Mucus layer forms a grease-like, soft chemical on the endothelial cell. Okay, endothelial cell, which secrete the acid, which protect us, which protect from the acid. Right? Some people who are now ulcerative. Okay. Oh, voice fine. So in a way, we have a bacterium. This protect the gut, and the acid resistant is, very well protected. So when you have a good bacteria, this is all happening. You see the left side. Whereas when there is no such bacteria, your endothelium is disturbed. Okay, so normal bacteria also goes into the intestine. Okay, it is called gut leakage. If bacteria goes inside, that leads to many other problems, right? Including enteric fever, right? So this is how dysbiosis brings many complications in the system. Next slide. You see, this complication comes when we jump into a food which is not ours. We eat something which is not to us. Okay, French fried rice is not a food. Cappuccino, okay, sandwich, bread with sandwich, it is not our food. So all that brings such alteration in the bacteria. That's what I wanted to convey. So it altered the hemostasis, meaning I believe you all know, a chemical, a substances. Stas is static, it comes from static. So we have a certain chemical in the system which are maintained stably, which maintain the healthy status. When such chemical goes down, a normal mechanism is halted. So you don't have a good bacteria in the system, you don't have any mechanism regulated. All the regulation gone. Imagine, we have line of control, people, our civilian soldiers are there. Okay, they protect from the invaders. If at all that peoples are removed by somebody or they die by themselves, what happens? You cannot believe, you cannot imagine. A moment is enough to destroy the total nation. Right? People are waiting for it. Same way we have a bad bacteria which can disturb total metabolism. In a way, we have a good bacteria which protect, which prevents such danger. Next. So this biases comes from the junk, which is not a nutritious, which is the nutrient for the bad bacteria. Very right informations are available, which are all the nutrient for the bad bacteria. Now, we could also observe the kids, small kids, who are very fond of, chocolates and sugary, candies. They always demand for it. You can check them. If there is such kid, they will be lean and, unhealthy. If they take the such hand for a longer period, they will be unhealthy. Reason is they have a bad bacterium in large number for a longer period. So that have disturbed their good health. That is important information. Next. So there are informations when you have such dishes in front of you. Dopamine and ghrelin, there are hormones which stimulates the hunger. You will not feel satisfied because the satiety will not take place. Satiation means what? I am satisfied. So I had morning idli, which is 3 in number. You will not believe. Such a beautiful, delicious food we have, which will bring satiety in enough number, right? If you take French fries or food like this, you will not get such satiety even after having 10 number. Okay, so as I said, it's not good for health. Importantly, next. You see, I'm eating, sir. What is the matter? Your gut is cause for every other disease. If you have a right gut, healthy heart, you will be healthier. If at all it is altered by your wrong food habit, then all the disease will come into play. Not only food, mental health also get disturbed. You become more anxiety. You are depressed. Reason, you don't have a right hormones produced in large number. Serotonin. You can Google it. Serotonin. Very important hormone, which encourages you, keeps you happy ever. But that is not there when you eat such chemicals. Serotonin comes from the protein. All that what I showed you is not rich of protein, which rich in calorie but no nutritive value. Nutritive value is slow. It is not in the right order. So IBD, okay, irritable bowel disease. Many people I would see, even I have a son who is studying, engineering. Immediately after meal, he rushed to the restroom. One day I was, following him. Hey, what is happening to you? Incoming, outgoing, there is no process. There is some problem in the gut. I've taken his fecal matter given for whole genome. I found there is a lot of Corynebacterium. Now his gut is resetted. He is having a healthy gut. I'll tell you, youngsters, if your food is not staying in the gut for more than 6 hours, you will not get the nutrient. It is waste. Instead of putting here, you can directly dump into the, your latrine. No use of taking it. It is waste of, enzyme, energy, all that. It must stay at least 6 hours. It has to be hydrolyzed. It has to be pyrolyzed. It has to be, absorbed by villi and should be processed and given to the construction, metabolism. If it is not taking place, it is purely utter waste of taking food. So one need to check it. Next. So all that. So next 6th slide, I wanted to reinforce what we are. How the good bacteria helps us. How to protect them. What food will nourish them. So ultimate, health rely on protecting this good bacteria. Right? Very important, right? Very important. Protecting bacteria matter to live a long life, right? So you see, microbes influence everything, right? Your appetite, efficiency of food usage and impact of immune system. Your immune system is primed. Immune system is trained. Immune system is improved by gut microbes. Our immune system is learning from microbes. T-reg you could have learned. Otherwise you can also refer. T-reg, T-cell population, which is orchestra manager here, which beautifully maintain the immune system. What are all way it should behave, right? It is the one, a commander, commands immune system. This is how. This will be possible when you have right bacteria. Otherwise, you don't have an efficient immune system that have oxygen, right? Imagine you have a security at home who's mentally not ill. You've given him a gun. When you come, he shoot at you. Imagine. That is how our own immune system will kill us. It is called autoimmune system, autoimmune disorder. This autoimmune disorder comes when you have a bad bacteria more and more. Because your immune system is not friendly trained, right? In a way, we are what microbes we have. See, my body cell number is 10 to the power of 13. What would be the number of bacteria in my body? Anybody can? You can tell, even if it is a wrong answer, no issue. My body cell is 10 to the power of 13. What will be the number of bacteria in our body? Certainly less or high? Yara solunga. Jastiya irukuma, kammiya irukudha? Tenfold higher. The bacterial load is 10 to the power of 14. Tenfold high. அப்போ இதில் யார் ஜாஸ்தி, யார் நாட்டாமல் பண்ணணும்? We do our egoistic way of dominating them. If we live by our way, then we have to be ill. Imagine. I-L-L என்னவா இருக்குது? இல். I-L-L என்னன்னா இல். If I live, I ill. V include my good bacteria. V-E remove L. Remove I. W-E-L-L. What happens here? Wellness. Wellness comes only with bacteria, not by I. I comes by that is my illness, imagine. So my ness is illness. When I am with good bacteria, it is, great story changer. Next. See, it is such a huge number of publications. All your behaviors directly influenced by gut bacteria. All your behavior are influenced by the gut bacteria, right? There are many experiments. A pilot study have been done, right? So it is proven what bacteria you have, that is your mood, right? Because it produces serotonin, dopamine like substances which keeps you happy, right? Happy hormones, it's all happy hormones, right? It keeps you happy. Next. Gut bacteria, okay, gives you the mood. Okay. So when you are not having a gut bacteria, you try to work on it. Right? You should have more of lactobacilli like aceri and bacillus cassovae, and there are clovians, bacillus clovians as well, bacterium like bacterioids groups and alpha diversity. All that plays major role. Next. A branch, right? Branch of study which deals with it. A bacteria which influence your psychology is psychobiotics. Psychobiotics. Right? We have different studies. Right? Next. Very interesting. So what you see in the black color is your DNA, your genetic material. Whatever you see in the left side, beautifully colored, all is your microbes. You have a diversity of population. This diversity makes you different. Okay, it is not by the genetic material. Okay, the diversity of the bacteria you have harbored since your birth is designing your texture, skin texture, designing your behavior, designing your health status. It designs everything. It designs everything, right? See, we are living in Namakal. If at all a cattle is not well, they would take, the fecal, cow dung of the healthy cattle and give it to the ill one. What is that? FMT, it is called FMT, fecal matter transfer. They were doing it. Till now, we are doing it, right? So, such a beautiful way one can bring flawless health, a good health, by having a right bacterium in the system. Next. So, this is what I wanted to reiterate. We have almost 99% of the gene functioning from the bacterial gene. All my metabolisms are borrowed. It is supported by the bacterium, which is there in our system, we have harbored. Right? Though we have genes which are predominant, but all the functions are all by the microbes. Microbial genes present in the system, right? So the right picture, what you see, human genes are in the toe, just a toe, right? Imagine. Rest are all by the bacterial genome. All the metabolism, if you take glucose, the TCA cycle, or amino acid metabolism, bifidobacterial pathways, all that is borrowed from the bacteria. That is how when you take a broad spectrum antibiotic, you wipe off all the bacteria, you find no metabolism taking place in your system. And you will feel almost, loosen some half of the cage weight. It's not so. Right? You lose some 4 or 5 kg of biomass. But obviously, when you don't have bacteria, you will feel almost 50% of your weight is gone. Right? In a way, we have such thing, which is also there in the reference. Next, little quick, right? You see, These microbes keep on changes. What we have in the infant is just opposite when we become elder. Imagine there is a pink color of bacterium, your firmicutes. The firmicutes is the one which goes with the aging, wrinkle in the skin and eating away all the muscles and you become very thin, right? That is due to the firmicutes. But in the middle age, it does work in a different way. All the energy it takes from the system and put it somewhere in the adipose tissue. Okay, adipose tissue. A waist size, waist circumference is higher when we have more such adipose tissue. That brings a lot of issue. So I will quickly run. I can read Madam's mind, right? So another 4-5 minutes. Is it okay, ma'am? Right. So being a teacher, this is a great help, right? So next slide. I quickly come, next slide. Next. Yes. Yeah, important slide. Okay. That alone is enough to convey what we supposed to learn. When we eat plant-based protein, which is highly nutritious, less calorie, very important, less calorie, but more nutritious. It helps in the enhancement of good bacterium like Bifidobacterium and Lactobacillus. These two are very, very key, components, bacterium, which helps in the keeping us healthier. Right? You see here, this Bifidobacterium and Lactobacilli produce more of short chain fatty acid. Short chain fatty acid, next slide I'm just having it. 3 short chain fatty acid. Okay? When this short chain fatty acid in a right order, your health status in right. You don't have any cardiovascular disease. You don't have any inflammation. Your health is in a right order. You have a strong immune system which always favors the good health. Imagine when you have animal protein, everything ulta, it goes opposite. You have more of trimethylamine oxide, less of short chain fatty acid. This is what I can say. That leads to increased cardiovascular disease and inflammation. Right? So, next slide. So, what we need to understand, you see, I am not saying this is the report which was published in 2018. Effect of dietary fat and gut microbes. Okay, dietary nutrients are less fat, more of nutrient, improves good bacteria, it nourishes gut bacterium, right? So less of cardiovascular risks. It is done, next slide. It is done with 45 participants. The participants given fibers, okay, prebiotics and fermented food have enriched Bifidobacterium and Lactobacilli when compared to the people who are taking less of this plant-based nutrient. Next. See, when we had this problem, we must understand. So this is the evolution of human being. The first cuminans had a purely vegetarian diet. Next, the omnivorous human, who's erectus, had a hunting habit. From there, this issue started. It is a story. I'm not saying there are information in the papers. Next slide. So this also says, right, when we had this problem, almost 3 million years ago, we were very, very normal, or get us very quiet, the healthy status. Next slide. See, this you can see till date. Okay, there are now changes in the food habits, right? Since from the first, meat eating, and we have changed it lot. Now we have a 3D food printing machine, right? We have lot of food textured to attract people. More and more attraction, less and less truth that we have to identify, right? Whichever food which attracts you, there's a danger inside. Okay, it is like fishing in the network. Next. So I can stop here, no problem. Ancient human had very healthy diet, which is purely plant-based, whereas the modern human like us, we eat almost by 10:30 night. We sleep before it get digested. It stays for a longer period undigested and become a food poison. That brings many other inflammation in the system. So in a way, I would conclude here by recommending a right food for healthy gut, which will maintain Bifidobacterium lactobacilli in large number. I have more on slide, which speaks about list of prebiotics. Very nutshell. You must take plant-based food, especially fiber-based food, right? Next important thing, you can have nuts and also food like which digest easily. It should not stay more than 4 hours. Okay. Next. You see, the industrialization and modernization lost alpha dibacterium. Alpha diversity is a good bacterium, right? By that we lost almost good bacterium in large number. Next, a few more slide. One, next. Yes, we have profile, which are all the bacterium helps, right? Faecalibacterium produce, the butyric acid. You see here the red color 2 star, the bacterium which produce more of butyric acid. Butyric acid is the one which enhance the metabolism, maintain good health. The another important short chain fatty acid we must have less and less is propaneic acid. Propanate, if at all you have more propanate, you are vulnerable for cardiovascular disease. Right? Next slide. Yes, I can stop here. So fruits, vegetables, seeds, nuts, whole grains, legumes, which are the one which enhance putrate protection, right? If at all you wanted to check your health status, you look for putrate in the fecal matter. That's enough, right? If you have more such compounds, you are in a healthy status. Next slide, another important. Next. So I have next. You see, there is a pathway, ISD, okay, a mutation in that pathway, which is one which controls glucose uptake, is mutated. That mutation grants you 430 years, imagine. 500% you can increase your longevity. So people who takes less and less of carbohydrate will live long. That is the one key and in take home message you can, right? So a mice model which is mutated for glucose metabolism, so all living for more than 5 years. This you can attest, you can note down, you can Google it, right? It is there in the internet. Next, important slide. Next. Yes. So when you have the prebiotics, which is your nutrients, when you have a right bacterium, which is your probiotics, these two produce a beautiful output that is called postbiotics. Postbiotic is your short chain fatty acids or this medium chain fatty acid and middle range fatty acids. All that play a major role in keeping you healthy, right? So researchers have gone to a state to produce more of postbiotics. Sir, என்னால் ப்ரீபயோட்டிக் எடுக்க முடியல, என்னோட பாக்டீரியாவை என்னால் மெயின்டைன் பண்ண முடியல அப்படின்னா you have a postbiotic as a capsule, which is costlier, but அதுக்கு alternate tendency எல்லாம் prebiotic-ம் prebiotic as well as probiotics you can take. You can make postbiotics, right? So I said stop here, have a healthy and a long life so that this nation will benefit from the research, the output from people like you all. Thank you so much for your patient listening. So few questions, if at all you have.
So thank you very much for your wonderful and fruitful lecture, sir. Please, any queries? The session is open for question. Participants, you please come forward and ask the question to our resource person. Any questions, any queries from participant side?
So I have a question like an origin of microbiome. So there is a theory that suggests that the microbiota is shared from our mother womb. Another theory suggests that the microbe cannot pass from the mother's womb to child. So what is your opinion in this?
Beautiful question. We must congratulate the gentleman. Your good name? Nithyendran. Adila konja Nityam. See whether this bacteria has come from mother or we ourselves harbored it. No chance for a mother to contribute. The place where the embryo is developing is sterile. Okay, there is no chance. It harbors microbes as soon as it touches the earth. It slowly builds. So that is where your habits matter. Sir, எனக்கு பல்லு விளக்க பிடிக்கல யானையெல்லாம் பல்லு விளக்குதா அப்படின்னா you cannot, right? So you don't maintain good bacteria, right? So all by your habits, all by your nutrition, all by the way you nourish this great body, holistic body, you harbor it. Okay, it is purely, by culture, tradition. That's why I reimpose it. Your tradition does lot. Your tradition contributes a lot. Okay, for every small things we have a reason behind it. We have to identify it. Thank you.
I take this privilege of giving wholehearted thanks to our Dr. A. Murugan sir, Professor and Head from Department of Microbiology, Periyar University, Salem. The way you handled the seminar was very appreciable and requires thousands and thousands of applauses, sir. Thank you very much. Once again, I express my deep-hearted thankfulness on behalf of our management, Director, Principal Sir, Organizing Committee members, our faculty members and my dear students. Thank you very much, sir, for your gracious presence here. Next technical session going to start now. Now, I'd like to take this opportunity to invite our next honorable invited speaker, Dr. Ashok K. Pandurangam, Associate Professor from School of Life Sciences, B.S. Abdul Rahman Crescent Institute of Science and Technology, Chennai. Sir had acquired BSc Biochemistry from University of Madras, MSc Biochemistry from University of Madras, MPhil Biochemistry from University of Madras, and PhD in Biochemistry from University of Madras. Sir's professional experiences, he has been worked as an associate professor as a till date. He has started his career as a postdoctoral fellow from Center for Cancer Research Institute, Children's Hospital Research Institute, California, USA, from May 2010 to August 2012. Postdoctoral fellow 2 from August 2013 to March 2013 from Department of Nutrition and Dietetics of Faculty of Medicine and Health Sciences, Malaysia. Visiting research fellow from April 2015 to August 2017 from Department of Pharmacology, Faculty of Medicine, University of Malaysia, Kuala Lumpur. And Sir, Associate Professor from August 2017 to till date from School of Life Sciences, B.S. Abdul Rahman Crescent Institute of Science and Technology, Chennai. Sir had secured so many awards. A few are received Best Faculty Male Award from B.S. Abdul Rahman Crescent Institute of Science and Technology, and as well as Sir is listed as a top 2% scientist in 2020, 2021 and 2022 in the field of oncology and toxicology by Standard University, USA. It's a great monumental achievement from the Ashok sir. Please give a round of applause. Received the Professor Radha Shanmugasundaram Proficiency Award for Best PhD for the year 2011 in the University of Madras, as well as awarded Senior Research Fellowship Yashaf by the Council of Scientific and Research in New Delhi, India. And as well as the Sir's publication goes on, and that is research and review article is 65, book chapter is 25, and book is 8 as an author. Thank you, sir. Knowledge is silver among the poor, gold among the nobles, and jewel among the princes. Here we have such a knowledge gentleman, and that knowledge competes him for the welfare of the student community. Let me invite Dr. Ashok Pandurangan, sir, Associate Professor, to deliver a lecture on the Portugal of gut microbes in the regulations of cold-associated cancer. Thank you very much, sir. Before that, I call upon Dr. Sudhagar sir to honor Dr. Ashok Kumar sir.
Okay. So, good morning, all of you. I'm so happy from the morning to see this much of crowd, even in this rural region. See, people used to come attend for conferences in and around Chennai. That is understandable. But there are so much of people, they used to come all the way from their natives to this place. That is so amazing. I think you can applaud for yourself for attending this conference. Yeah, it's good. Thank you so much for inviting me for this conference. First, I would like to thank the management for organizing this kind of conferences so that we can meet a lot of old friends here. And I would like to thank my good friend, Dr. Sudhakar. We were like friends more than 20 years. So it is like a nostalgic moment for us to meet. Also, one of our friend is here, Dr. Kumaran. We were all together studied in our PhD during our PhD time in University of Madras. Before I go into this topic, I could admit one thing: I'm not a microbiologist. To be honest, I'm not a microbiologist, but the disease what I took, what I studied for more than 15 years, the colitis-associated cancer, it has certain relevance with gut microbes. So I feel like it is better to start with an overview about gut microbes, and I'm going to tell you what kind of research I have done, some of the results which I have done in my lab, but I'm going to discuss with you. If you have any questions and all, you can raise your hands and you can interrupt me in between. Otherwise, if you have any other questions, later also you can ask. Okay? Next slide, please. So, gut microbe. Do you know how many microbes, what number of microbes that live in your gut? Do you have any idea? Hope most of you are students. Do you have any idea how many microbes or what number of microbes? Just a guess, wild guess. It may be wrong or it may be right. Until I read this particular topic recently, I also have surprise. Next, please. So 100 trillion microbes that live in your gut. So you know the gut where it starts from. It starts from your mouth to your anus. In this region, there are 100 trillions of microbes that live in your gut, right? They are not harmful to you. They are beneficial. That is the important part. All these gut microbes, they are not harmful to you. They are beneficial, right? Next slide, please. Oh, okay, it's okay. Go back, please. Fine. Next. Okay, so what are the organisms collectively we call gut microbes? What are the organisms that live means bacteria, virus, fungi, archaea, as well as eukaryote. I'm going to tell you some of the facts before I go into actual topic. So when you weigh these microbes, 100 trillion microbes, that weighs about 2 kg, right? And when you calculate that, equate your number of cells present in your body, that is 50% of the cells which corresponds to gut microbes. Okay. Because of these microbes, they form a kind of complex ecological system in your gut, which it is not fully understood. Very little was understood. That too, whatever is understood, it is an ocean. It is not completely understood, but still it is little understood. Whatever it is understood, whatever the benefits that it does to you humans, that itself a ocean kind of knowledge is there. Okay. And there are 1,000 variety of these microbes are there. That is another fact. Okay. And also when you compare with the entire human genome, there are 150 times more genome. The human genome, you know about the vastness of a human genome, right? But when you compare to the human genome, there are 150 times more. 150 times is multiple. You multiple 150 times. Human genome is 1. They are 150-fold higher. Imagine the vastness, right? So, these gut microbes, they form a symbiotic relationship with human host. So you know what is symbiotic, right? They help each other. The human host helps the microbes, and microbes helps human. Let's see how it helps. Next slide, please. So what is their vital role in human means? They help in metabolism. They provide nutrition. It involves in immune functions. I'll show you some of the slides next. They also involve the physiology and prevention of diseases. They try to cut off the pathogenic bacteria. They try to prevent the pathogenic bacteria, right? So all these functions, right? So look at all these things, 7 functions which is already mentioned here. Start from protection against pathogens. They synthesize vitamins in the colon, right? Vitamin K and B12. It's also involved in the immune system development. It also helps in the immune system development, right? The vital function is it promotes angiogenesis in the intestine. Angiogenesis is nothing but the formation of new blood vessels. That also can be facilitated by this gut microbes. Also, it helps to store the fat. Then it also helps to produce short chain fatty acids. These SCFA, that is short chain fatty acids, they help in regulation of immune system. Okay, also helps to digest the fiber materials that you intake, right? It also, interestingly, it also modulates your nervous system. Your nervous system is a master regulator of any human being, right? So it has an impact in regulating nervous system. Next, please. Okay. So these are the ways that these gut microbes help in regulation in your body, right? So it helps to digest your breastfeed milk, even in the smaller child, the baby. This particular bacteria helps to digest your breastfed milk. And also it helps in digesting the fiber material that you intake through your food. The complex fiber materials, excuse me, to avoid cancer. The colorectal cancer is mainly, one of the cause was less intake of fiber and high intake of cholesterol. So this particular bacteria, this particular gut microbes can help to digest the fibers, so to avoid the cancer risk. Then it controls your immune system. I'll show you one specific slide how it controls the immune system so that you will understand, right? It also controls your central nervous system, right? Next slide, please. So I've given some of the microbial chains that are stays in your gut. Starts from your stomach, you have lactobacillus, streptococcus, staphylococcus, enterobacteriaceae, duodenum, and jejunum. See, these are all the intestinal parts, different parts of intestine, duodenum and jejunum. Your intestine, whole intestine will be, small intestine will be separated into duodenum, jejunum, and ileum. Ileum will be closer to your colon. Duodenum will be closer to your stomach. After stomach, until colon, you have variety of bacteria that stays in your small intestine and colon. See, look at their axis. It connects all the major organs. Starts from your brain, your endocrine axis, your heart, lung, liver, spleen, bones, muscles, even in skins. Everything they control. These gut microbes, they control. Okay, next slide, please. Okay, so another important term that you must know is dysbiosis. So what is dysbiosis? How you define dysbiosis means there's an unbalance between this gut microbes, that is beneficial microbes, that is termed as dysbiosis. This is the one that directly related to the disease which I was working on, ulcerative colitis and colitis-associated cancer. Next slide, please. Okay, dysbiosis, I already told you, that is imbalance between the good bacteria and the bad bacteria. That is pathogenic bacteria. Okay. So normally what happens is, directly this particular dysbiosis is related to ulcerative colitis and colitis-associated cancer. That is CAC. CAC means colitis-associated cancer. So what they did was, the scientists, a group of scientists, they isolated fecal bacterial DNAs. Sorry. From that, they identified some of these bacteria, they involved in the pathogenesis of ulcerative colitis and colitis-associated cancer. So you can do this very easily now. You can isolate the DNA from, fecal material of any cancer patient or an ulcerative colitis patient. And then you can just sequence that using next generation sequencing. So that will give you an idea what kind of bacteria that is highly dysregulated or what kind of bacteria that is present in the species that helps directly into the pathogenesis of this disease, right? Also, some of the studies already said that bacteria can promote CAC progression, that is colitis-associated cancer progression. One group of people, they are doing research on what kind of bacteria they promote colorectal cancer. A Chinese group was working in a Shanghai laboratory. They are showing that ingesting these probiotics to these animals which have ulcerative qualities that can cure your ulcerative qualities. So it's a kind of two-edged sword. Some group of bacteria, they promote ulcerative qualities and colitis-associated cancer. Some group of bacteria, they cure, they act as a cure, right? So it is a balance. Next slide, please. So this is the picture I just want to show you, how a normal probiotic that acts, that regulates in the normal cell in the colon, as well as during the colitis-associated cancer, how they regulate, right? So this is normal probiotic colony, you see. This is your villi, colon villi or intestinal villi. Most of these bacteria, they promote apoptosis. They try to inhibit inflammation by controlling all these inflammatory cells, especially macrophages, neutrophils. All those cells will be dendritic cells. All these cells will be suppressed, or it is downregulated by these probiotics, right? In turn, it inhibits all these cytokines that are released by these immune cells, especially macrophages, natural killer cells, right? You see, TNF-alpha, interleukin, all are very crucial pro-inflammatory cytokines. They are all downregulated. Inducible nitric oxide synthase and COX-2, cyclooxygenase 2, those two were very important inflammatory mediators. Those are all downregulated by these probiotics. In turn, it promotes apoptosis. It activates aspases, right? So that the apoptosis will be facilitated. On the other hand, this tumor suppressor gene, P53 and all, which is upregulated. So the probiotics kept the cells intact and in the check. On the other hand, when you look at this colitis-associated cancer condition, see, all these, you see T cells are activated, macrophages activated. On the other hand, this interleukin pathway activated, that activate T cells and dendritic cells. All these immune cells were highly activated on one end. On the other end, related to that particular signaling, PI3K and NF-kappa B through microRNA 21 axis, everything will be dysregulated. This promotes tumor. Okay, when your gut microbes were disturbed, that is in the condition of dysbiosis. Next slide, please. Okay, so I completed my overview about gut microbes. Hope you understand a little bit gut microbes, how important it's to the host, especially to us. Okay. So the topic which I was working on, inflammatory bowel disease. So inflammatory bowel disease is defined as inflammation in the colon, generally, and inflammation in the colon. So that can be divided into ulcerative colitis and Crohn's disease. What is the difference between ulcerative colitis and Crohn's disease means? Ulcerative colitis is the colon completely inflamed. But in the Crohn's disease, it's a patchy. A smaller region of colon is affected, another smaller region of colon is affected, not completely affected, right? Next slide, please. Okay. So general etiology is so far unknown. What is the main causative means? The factors involved, environmental, genetic, and immunological factor. What is the clinical symptom means? It may be abdominal pain, diarrhea, mucus, bloody. People can develop all these symptoms, or any one or two symptoms can be developed. How you can identify this particular disease? What does the diagnosis means? Excessive activation of TNF-alpha, interleukin 1 beta, 1, 6, 17, and 21. All these pro-inflammatory cytokines were highly dysregulated, and the ROS, that is reactive oxygen metabolites or reactive oxygen species, they are all upregulated in the colon. This leads to the mucosal damage. Mucosal means you have a colon-like breastwater structure. Hope you know already. You would have studied in physiology. That will be completely erased in this particular disease. Next slide, please. So how to mimic this model in animal? That is a big challenge, right? So you want to develop a drug for any disease, you need to develop an animal model or an in vitro model to test your drugs against this particular disease. So scientists have developed a model, specific model, that is dextran sodium sulfate induced model. This is a polysaccharide. When you ingest this polysaccharide in the drinking water to animals, they develop exactly, they mimic like a ulcerative colitis. Okay, so you have to use specifically the molecular weight of that particular DSS is 36 to 50 kilodalton. Next slide, please. So what we do to induce acute colitis, the colitis can be separated into acute and chronic. Acute means within 6 days if you treat DSS, on 7th day the animal colon will be inflamed. Chronic means you can treat them with 2 or 3 cycles of dextrin sodium sulfate. So I used to study in the acute colitis model, where the mouse were treated with 3.5% of percentage of DSS in drinking water, treated for 7 days. On the 8th day, the animal get ulcerative colitis. Next slide. So what has happened when you ingest DSS to this animals means, on the first cycle of DSS, you see this is a chronic model. On the first cycle of DSS ingestion, that will increase all these inflammatory cells, especially neutrophils, macrophages, all the cells, not only these cells. All the inflammatory cells, they were upregulated. That in turn activate all these pro-inflammatory cytokines and cell adhesion molecule were upregulated. In between the second cycle of DSS, there is water treatment was given. That is, you withdraw the DSS so that all these cells all these signs of pro-inflammatory cytokines were downregulated. That is the recovery phase. We call it as recovery phase. Again, when you ingest the DSS, it will aggravate the disease. So all these mediators were upregulated in the chronic phase. Next slide, please. So, when untreated ulcerative colitis, what happens? Next, click. That will lead to the development of colitis-associated cancer. That is chronic inflammation. If you don't treat it, that will develop as cancer. Right? Next slide, please. So during the colitis-associated cancer, what are the molecular events that occurs? So in colorectal cancer itself, it is divided into spontaneous and colitis-associated cancer. Spontaneous means regularly people used to get because of their familial history or environmental problems, environmental exposure, or because of their genetic, they develop spontaneous cancer. So during the spontaneous cancer, what happens is, the event is almost same. Conversion of normal epithelia to carcinoma is same. So the normal epithelia will be converted into ACF, that is adenomatous, abundant crypt foci. Then abundant crypt foci will be converted into adenoma. Finally, it will become carcinoma. So the molecular event is completely different between spontaneous and colitis-associated cancer. You see here, only the APC, which is tumor suppressor gene, that is downregulated, and beta-catenin, which is upregulated from the Wnt signaling. During the adenoma stage, the KRAS is upregulated, COX2 is upregulated. Finally, in the carcinoma stage, the p53 and TGF-beta receptor is downregulated. They both are tumor suppressor gene. But look at this colitis-associated cancer. There is dramatic event that occurs. During the ACF condition itself, all these cytokines were upregulated. And NF-kappa B and STAT3, they both were very important key transcription factors. You know very well NF-kappa B is a central regulator of inflammation. And STAT3 is the junction or a niche between inflammation and cancer. All are upregulated during the ACF stage itself. Finally, in the adenoma stage, the NF-kappa B and STAT3 expression were multiple fold upregulated. And during the carcinoma stage, it's almost similar like spontaneous cancer. Next. I think you can skip next. Next. Okay. So we used to treat this particular boldin. Boldin is nothing but an alkaloid which is isolated from a natural product. The structure is given here. It has variety of pharmacological benefit, which has anti-inflammatory, antipyretic, atherosclerosis, anti-carcinogenic, anti-diabetic. There are multiple. It also tends to regulate very key molecular signals. Okay, that is the reason we chosen this particular drug to treat this disease. Next. So first study, we done two studies. One is we used this boldin to treat acute ulcerative colitis. And the second study, we used to treat this boldin to colitis associated cancer. We will see one by one. Very shortly, I'll finish up another 10 to 15 minutes. Next slide, please. So first experiment, what we did was, we take 3 group of animals. One particular animal, we treat them as control. The second animal received 3.3% DSS in drinking water. And the third group of animal that received DSS plus 50 milligram per kg body weight of Boldin for 7 days. After the 8th day, next slide please. We checked all these parameters: disease activity index, colon length, spleen weight, histopathological analysis, immune cells staining, and expression of immune inflammatory mediators. Next slide, please. So first we check the disease activity index. So disease activity index means we calculate during the time of ulcerative colitis. We check 3 different parameters: body weight of the animal, rectal bleeding, and blood in the stool. These 3 parameters we use to monitor every day. Next slide, please. So this is how we score for disease activity index. For example, we take 3 parameters, that is weight loss, stool consistency, and visible blood in the feces. So if weight loss is 1 to 5%, we give 1 scoring value. If it is 6 to 10% is 2, accordingly we give a scoring. And stool consistency, if it is normal, we give 0. If it is loose stools, we provide 2. If animal have a diarrhea, we provide 4 marks. Similarly, visible blood in the feces, if it is none, it's 0. If you see very slight bleeding, we will provide 2. If it is gross bleeding, we will provide 4. Likewise, we score for all the 7 days, and finally we plot this graph. Go back. Pinnala. So we plot this graph. During the DSS treated animal, dextrosodium sulfate treated animal, we see huge disease activity index because they lose body weight, they are seen blood in the stool, they are diarrhea. So all these changes that occur, because of that, the DSS value is higher. But subsequently, when we treated with Boldin, it tried to bring down this disease activity index, so it is which is significant. Also, we check the colon length. Why we need to check the colon length means, when you treat DSS, that will erase the brush border villi structure in the colon. Because of that, the colon will shrink. So that is an indication of ulcerative colitis is induced. So when we check the DSS, the colon length was shortened. You can see this colon length was shortened from DSS-induced animal. The normal control animal have almost 12 centimeters, but DSS-treated animals have 8 centimeters. When you treat this, it's not completely protecting the colon, but still it is significantly protecting. That will be around 10 centimeter average. Also, we check the spleen weight. Normally, when you treat it with DSS, because of the damage that occur in colon, where the bleeding will occur, to compensate the bleeding, the spleen cell tend to produce more blood cells. Because of that, the spleen enlargement will occur. So that is why in the DSS-induced animal, the spleen weight will be higher compared to the control. When treated with the DSS, the spleen weight was become normal. So this gave an indication that this Boldin is working fine. Yeah, we can go further to study the molecular events. Next slide, please. Next. We also checked the histopathological analysis, that is the microscopic analysis of colon. In the control, you see the very healthy brush border structure of colon. But you look at the DSS, this villi structure is completely erased. This is an indication that DSS is erasing all the brush border structure, right? The ulcerative colitis was induced. Also look at the DSS animal treated with Boldin. There will be a restoration of brush border structure even in LD. Next slide, please. We also confirm this using scanning electron microscope. The scanning electron microscope which scans the surface of the colon. We seen the complete damage of colon in the 500X. This was partially protected by the Boldin when you treat it with Boldin about 50 milligram per kg body weight. Next slide. Then we want to check what is the role of this Boldin on on immune cells recruitment. Usually immune cells recruitment is one of the key factor in ulcerative colitis. First we check the expression of CD68, which is a marker for monocyte and macrophage. When we treated with this immunohistochemical staining of CD68, we found a huge number of these brown dots that indicates the recruitment of monocytes or macrophages. But when you treated with Boldin, the brown dots were very minimal. And we also quantified it. That also provide the similar result. And we also checked the activity of myeloperoxidase. Myeloperoxidase is one of the marker of neutrophil. Okay, so the amount of myeloperoxidase directly leads to the amount of neutrophils recruitment. And we found in DSS animal, the myeloperoxidase activity is higher. Subsequently, when you treat it with Boldin, this brings down significantly. So this gave us a clue that Boldin directly controls the immune cells recruitment. Right, next slide. We want to check what is the status of these pro-inflammatory cytokines because they all play a very important role in activating. I will show you one slide how these pro-inflammatory cytokines, prolonged activation of this pro-inflammatory cytokine that will promote tumorigenesis. I will show you in the later slide. We checked 3 different cytokines by PCR, TNF-alpha, interleukin 6 and interleukin 7. Excuse me. All these cytokines were upregulated during the DSS condition. There is no surprise at all. In ulcerative colitis, that is one of the marker. And when we treated with Boldin, our surprise brings down very, very significant level. Right? This indicates Boldin controls the inflammation through downregulating the immune cell recruitment. Next slide, please. Next slide. Right. So, we also want to check in the in vitro condition how this Boldin is effective. So we took raw cells are macrophage cells, right? We treated these raw cells with LPS to activate inflammation at about 10 nanogram. And then we treated this LPS with 20 microgram per ml and 50 microgram per ml concentrations. And we checked the status of NF-kappa B. We also checked the Western blot level. In the Western blot condition, the LPS activated macrophages, they produce higher number of P65 NF-kappa B. And when you treat it with Boldin at higher concentration, dose dependently, it brings down the NF-kappa B. Similarly, we also seen in the fluorescent microscopic analysis, where you can see in the LPS, all these NF-kappa B were sitting in the nucleus. But when you look at this particular image here, all the NF-kappa B were sitting in the cytoplasm. This blue indicates the nucleus. The green indicates the NF-kappa B expression. So all these expressions were sitting outside the nucleus. That indicates NF-kappa B is inactive. NF-kappa B is active when it is sitting in the nucleus. It is inactive when it comes out of the nucleus. So that also we confirmed. It regulates NF-kappa B. It downregulates NF-kappa B. Next slide, please. Next. Okay. So with this experiment, we published this paper in the biofactors. Right? Next. Right. So we came to know that Boldin is very effective in ulcerative colitis. We want to extend our further studies to check whether this Boldin is control the tumor formation. So for that, we need to undergo colitis-associated cancer. Right? Next slide, please. So in this study, we took the same 3 groups. First is normal control. Second is AOMDSS-induced mice. So to induce colitis-associated cancer, single dose of, I'll explain the next slide. The third will be AOMDSS plus 50 milligram per kg body weight of Bodine was treated for 62 days. Next slide, please. So how to induce AMDSS induced colitis associated cancer model? So we follow this. So on the day 1 or day 0, we inject 10 milligram per kg body weight of azoxymethyl, which is a known carcinogen of colorectal cancer. It induces colorectal cancer on day 0. After 6 days of gap, for 7 days, we provide DSS, dextrose sodium sulfate, in drinking water, followed by 14 days, normal recovery period. This was repeated for 3 cycles. On day 62, the animal developed tumor in their colon. This is the shortest model to develop tumor in the animal. I have worked in many models. None of this model will provide you within 62 days tumor in the animal. First, we check the tumor number. Probably that slide is not working here. First, we check the tumor number. After 62 days, we check the tumor number. So the AMDSS animal got averagely 10 tumor per animal. But when treated with Bodine, it will become 6 tumor per animal. Also, we check the tumor size in those animals. We found that the smaller number of tumors, the smaller size of the tumor was very less when you treated with Bodine. Then we check the histopathological analysis of all these experimental group. You see the control, that is normal breastbone structure as usual. When you see this AMDSS animal, the whole region was transformed into tumor. When you treated with Bodine, the tumor size become shrink, very smaller. And this was confirmed by PCNA expression. We studied the immunohistochemical analysis of PCNA, that is proliferating cell nuclear antigen. That is a marker for cell proliferation. All these brown dots indicate the cell proliferation, higher cell proliferation. When you see this third, which is treated with Bodine, there is very comparatively less number of cell proliferation. That is an indication Bodine inhibits cell proliferation significantly. Next slide, please. As usual, we check the cytokine analysis. We found that AMDSS model have higher pro-inflammatory cytokines, TNF-alpha, interleukin 6, and interleukin 17. All the cytokines were tested through ELISA. And when you treated with Bodine, it all brought down all these cytokines significantly. So this is an indication Bodine is even effective in the tumorigenesis stage also. Next slide, please. Okay. So far we have found the Bodine is very effective in controlling the inflammation. But if you control inflammation alone, is it enough? Or you should act, what is the factors that contribute for the inflammation to cancerous stage? You need some niche to convert the inflammation to cancer, right? So that particular niche is STAT3 signaling. So STAT3 signaling is nothing but this GP132 receptor, interleukin 6, there is a pro-inflammatory cytokine which acts as a ligand for this GP132, which further phosphorylates JAKs. JAKs further phosphorylates STAT3. STAT3 is one of the important niche between inflammation and cancer. STAT3 signaling, please note it down. Okay. So the phosphorylated STAT3, when it enters into nucleus, where it bonds to SOX3, suppression of cytokine signaling 3, which activates all these genes. You look at these genes, survival genes, proliferation, angiogenesis, metastasis, adhesion, inflammation, all these genes were activated by STAT3. So it activates a variety of cell proliferation and survival genes. So you want to study what is the effect of Bodine on STAT3 signaling. 1 or 2 slides I will complete. Okay, next slide please. Right, I told you how this inflammatory cells, they further potentiates the normal epithelial cells, right? Through paracrine and autocrine mechanism. For example, let's say your myeloid cell or lymphocytes, they are activated, this particular STAT3. They produce all these cytokines. Those cytokines will further activate other neighboring cells. Let's say this is epithelial cells. So these produced cytokines, they activate their own signaling and that activates other cells also. So this is autocrine means they activate their own cells. Paracrine means through the circulation they go and activate other fibroblasts and lymphocytes or other immune cells. This is how the inflammation aggravates. Okay, next. So to check what is status of this STAT3, what we did was we treated excuse me, SCT116 cells, that is the colorectal cancer cells, with interleukin 6. Directly we added the cells that induce inflammation. Okay, to convert normal epithelial cancer to inflammation-driven cancer cell. When we check time-dependent manner, we found that in 24 hours when we treated with Boldin, the STAT3 was downregulated. There is no difference in the total STAT3 as expected. Next slide, please. We want to find out what is the actual mechanism. So for that, we tested the upstream of STAT3 was SH2. That is SH2 containing tyrosine phosphatase that regulates STAT3. So we would like to check whether it is SHP2 mediated. For that, we took an siRNA of SHP2 and we added the cells. So that indicates the SHP2 gene was silenced. After that, we dumped these cells with and without Boldin. Without SHP2, even if you inhibit SHP2, the STAT3 was downregulated in Boldin. This indicates this STAT3 downregulation of Boldin is mediated through SHP2. Next slide, please. Okay, next, click. So when you treat DSS, that will activate TNF alpha, that further activate interleukin 6. Click. So interleukin 6 bound to this JAK that activates STAT3. When STAT3 enters into nucleus, that activates variety of gene. In other turn, DSS directly activates NF-kappa B. Through NF-kappa B, inducible nitric oxide synthase and COX-2 were activated. Next. But when you treat it with Boldin, it controls TNF-alpha, it controls NF-kappa B. Through controlling NF-kappa B, it inhibits Hynas and COX-2. It directly controls the translocation of STAT3. So when the STAT3 is not translocated into the nucleus, which will be no tumor formation. Next. So these are the list of publications that we published in this field. Next. Okay, time to thank my superiors. This is the first, my postdoc lab, where I worked in Children's Hospital Research Center. Here, I learned this colitis-associated cancer. It's a new type of cancer, where I started work in this lab. So she is my boss, Dr. Juhi Sabha. Next. This is my second postdoc lab, which is in University Putra, Malaysia, Dr. Narayan Mohammad Esa. And they are my PhD students. Next. This is my third postdoc lab, Professor Muhammad Rais Mustafa lab. So he is my boss. He's head of pharmacology. These two people are assistant professors. These are all PhD students. And he is a visiting scientist from Morocco. So very nice place where I worked. Next slide, please. And finally, this is my lab. I have 3 PhD students. They are working under me now. So Pratibha Ashwati and Mohammad Kashif. Next. This is my contact information. If you have any queries, you can contact me. Next slide. Next. Thank you. If you have any questions, you can ask.
Good morning, sir. Myself, Hitendra. So I have a question that you have selected bold in, right, sir? So how you have selected and how you have allocated?
See, we have a pharmacology department in association with faculty who is working on it. So it's very fascinating to work with them because they commission a record. So they go to all the they collect different plants. From there they bring to lab. There brings Okay, last, I'll just complete this picture, the history of this picture. Do you recognize this picture? Do you recognize this bridge? No. How many of you saw Varnam Ayiram movie? So this is very famous Golden Gate Bridge that connects San Francisco and Sausalito. Every talk where I go, I used to give a brief history about this picture. A boy from a small village, right? Not even 1,000 people were there in my village. So I could able to go to this bridge, right? Stand next to that bridge, I took a picture. Why don't you, right? Definitely you all have a chance to go, right? Do a PhD, to have a better future, right? You can go and visit places like this and inspire as many as students in future, right? Thank you so much.
Sir, please speak. Thank you, sir. Thank you very much for your motivational and inspiring lecture for our participants. Please be silent. On behalf of our management and organizing committee members, faculties and participants, I wholeheartedly thank Dr. Ashok sir from Chennai for giving a motivational and inspiring lecture. Thank you, sir. Thank you very much. Now we can have a tea break. Before tea break, the oral presentation session is going to start at 12:15 p.m. Oral presentation session is going to start at 12:15 p.m. at B Block Mini Seminar Hall. So today, the oral presentation number 2 to 90, oral presentation 2 to 90 numbers are going to present your presentations in the B Block Mini Seminar Hall. The list has been displaced in front of that B Block Mini Seminar Hall. Oral presentation இன்னைக்கு யார் பண்றீங்களோ அவங்களோட லிஸ்ட் எல்லாம் டிஸ்ப்ளே பண்ணிருக்கோம்ப்பா. If you have any doubts relating this, you please come and approach the MOC desk. இங்கேயும் லிஸ்ட் இருக்குங்க. இன்னைக்கு oral presentation யாருக்கு என்ன டைம் எல்லா லிஸ்ட்டும் MOC டெஸ்க்ல வச்சிருக்கோம். You please come and ask the thing. Afternoon, after lunch break, you are having poster presentation. Those who are involved in poster presentation, you can display your posters in B Block 007. B Block 007 hall numberல உங்களோட posters lunch breakல display பண்ணலாம். Remaining students, for after tea break, 20 minutes you will have a tea break. Tea break அப்புறம் remaining இருக்கக்கூடிய students எல்லாம் you please assemble in this conference hall at 12:10. ஓரல் ப்ரெசன்டேஷன் மார்னிங் யாருக்கு இருக்கோ அவங்க மட்டும்தான் அந்த ஹாலில் அலோவ் பண்ணாங்க. சரியாப்பா? ஃப்ரெண்ட்ஸோடு சேர்ந்து அங்கே போய் உட்காந்துடக்கூடாது, நான் சொல்லிட்டேன். யார் ஓரல் ப்ரெசன்டேஷன் பண்ணுறாங்களோ அவங்க மட்டும்தான் அந்த ஹாலில் இருக்கணும். ஸோ இந்த லிஸ்ட்டில் யார் யாரோட நேம்ஸ் வரலையோ அவங்க எல்லாமே இந்த லிஸ்ட்டில் அப்டேட் பண்ணியிருக்குப்பா. எல்லா ஸ்டூடெண்ட்ஸோட ஓரல் ப்ரெசன்டேஷன்ஸ் லிஸ்ட்டில் அப்டேட் பண்ணியிருக்கோம். ஸோ அதனால் ஓரல் ப்ரெசன்டேஷன் இருக்கக்கூடிய ஸ்டூடெண்ட்ஸ் மட்டும்தான் நீ பி பிளாக் மினி செமினார் ஹாலுக்கு போகணும். ரிமைனிங் ஸ்டூடெண்ட்ஸை நீங்கள் ஸ்டாஃப்ஸ் அலோவ் பண்ண மாட்டாங்க. ப்ளீஸ் பி இன் திஸ் கான்ஃபரன்ஸ் ஹால் after a tea break. Okay, so this is an information. So now let us have a tea break for 20 minutes. You please assemble back at 12:05. Another one, the world presentation. I request Dr. E.G. Wesley sir to judge the oral presentation today at B Block Mini Seminar Hall. As well as I request Dr. Kumaran sir. Sir, welcome sir for our session here. Dr. Kumaran sir from Department of Botany, Government Arts College, Arur. Welcome sir for our conference. I request Dr. Kumaran sir and Ashok sir to judge the poster session afternoon. Okay, thank you. Tea has been arranged near the conference hall. எப்பயும் போல அங்கே போயிடாதீங்கம்மா, இங்கே கான்ஃபரன்ஸ் ஹால் முன்னாடி தான் இருக்குது. சீக்கிரம் டீ சாப்பிட்டுட்டு சீக்கிரம் வந்துடணும். ஓகே, ஹாவ் டீ பிரேக். தேங்க்யூ. I request all our participants to assemble quickly. Very shortly, we are going to start our third technical session of this day. Once again, I welcome you all for our third technical session of this day. I wholeheartedly welcome our next honorable invited speaker, Dr. P.K. Sendilkumar, Assistant Professor, Department of Microbiology, Annamalai University, Sidambaram. I'd like to take this opportunity to introduce our next honorable speaker. Sir has acquired BSc Microbiology from our institution. He is our proud alumni. As a batch of 97 to 2000 at BSc Microbiology at Muthayammal College of Arts and Science, MSc Microbiology in the year 2002 at JJ College of Arts and Science, Pudukkottai, MPhil at 2003 at JJ College of Pudukkottai, and PhD from Annamalai University. Sir had started his career as assistant professor at Annamalai University, and he has been elevated as assistant professor and head in charge at Annamalai University, and as well as DOE coordinator in microbiology as an Annamalai University, and now he is sitting as an assistant professor and head in Annamalai University. Sir had produced 8 MPhil scholars and 9 PhD scholars under his able guidance. Sir is a board of studies member in Madras University in microbiology, both UG and PG, NGP Arts and Science College, AVC College, and St. Joseph College, etc. Sir has secured so many awards. A few one is Best Performance for National Ninth Day from Annamalai University in the year 2015. UAE Indo-Louis Pasteur Students Excellence Award of 2022 at International Multidisciplinary Research Foundation from 2022. And as well as Sir has published more than 15 numbers of papers in national and international journals with high reputed impact factors, and as well as book chapters, and as well as conferences, seminars. And Sir has been completed so many major and minor projects in the grant sponsored by UGC, DSCR, CSCR in the more than 20 and 25 lakhs, etc. Thank you. The true man has no worries. The wise man will not be puzzled. And the brave man has no fear. I genuinely invite an experienced, skillful personality, our proud alumni, Dr. P.K. Sendilkumar sir, Department of Microbiology from Annamalai University, Chidambaram, to deliver lecture on discovery of novel drugs from marine actinomycetes. Thank you, sir. Before that, I would like to invite our director, sir, Dr. R. Selvakumaran, sir, to honor our chief guest with a shawl and a shield. Welcome, sir.
Honorable management authorities, principal and organizing secretaries, my well-wisher and my good teacher, Dr. Anbagan sir, and research director, Sudhagar sir, Sagita ma'am, then my dear friends and my seniors, Selvam Anna, and fellow colleagues and students from same college and various colleges from Tamil Nadu and out of Tamil Nadu. Good morning, every one of you. Really, I am very much proud and pride before standing you as a resource person as well as a old alumni of this great institution. So why I am telling this? When I was a student in 97, joined here as a microbiology student, really I don't know how to, I'm not interested in that subject, microbiology. That is very much important. So, why? Because I came from pure Tamil medium. So, our syllabus will be only totally we need to convey all the things by English. So, Wednesday class started. So, first teacher came and explained well about what's meant by microbiology and their scope. I am not interested. So, really I am seated in the first bench. So, I am not willing. So, I decide myself, I quit on Friday. So, only 2 days permitted myself. So, we have to wait for 2 days. Then we quit. Then we get apology from father. Then we joined BCom or some other course. Like that I decide. Then second day also, I feel same things. But third day morning, really Anbalagan sir came and explained well the subject microbiology and their impact by Tamil. Really then I have confident. So little bit confident. Yes. So little bit confident. So I could complete this course like that. Then that time one more professor came and explained about virology. Then I am interested in virology. So that day itself, I studied well. I didn't get any arrear because of this institute. Now I elevated myself as a assistant professor in permanent position. Okay. So this is the information for you. Why? Because even though not interested, due to pulled us by our parents, sometimes we need to join any particular course according to their interest. So even though, how come we rule ourselves in that particular field, definitely one day it will bring us in, what we need to achieve. Definitely we will achieve that position. So that is, I want to present here. Then today, I'm going to discuss about what is meant by drug, antibiotic, then why particularly I'm concentrating on actinomycetes. So I'm going to present my topic. Before that, we are celebrating two things now. I think 2 days over. Am I correct? So 1.5 days over. So you are plunged with lot of knowledge regarding so many persons already delivered a lecture. And simultaneously you are celebrating that cricket. Am I correct or not? Okay. So Kohli break the record, world record already. Who did, Sachin record. So am I correct? You are enjoying or not? Some of you now also you are enjoying, I think. Am I correct or not? Unmingla illaya? Whether you may reach me or not. Okay? So that is very much important. So study is not only enough to, explore us. Okay? So along with academic, we should know what happening in and around us. So how the world exploring what things, that is very much important. So regarding that, so we should focus cricket. Am I correct? And gazettes. So most of you in last bench, now also I'm watching, most of you, your fingers are typing something. Am I correct or not? Whether sir tell the true message or not, like that there are somebody searching, whether Kohli break what, like that I think you are fetching the message. Am I correct or not? Okay. So then, you are actually you are most probably you are now being MSc and few of you may be UG students. Okay. So you know what is meant by disease and you are currently what type of antibiotic or drug you are consuming. So you know the difference between drug and before entering the subject, just I want to clarify. Okay. So drug is a chemical substances, any chemical substances, so which may normalize our physiological function. That is called drug. Example, acetaminophen. Now we call it as paracetamol. Okay. So antibiotic is a chemical substances are synthetic or semi-synthetic derived from natural source or plant. Natural source means plant or herbals or some other molecules. And that would be act against other living thing. Okay, that is called antibiotic. Sir, these two things are nowadays, and nowadays we are facing so many problems how to handle this antibiotic and drugs. Am I correct? So you know all the, you know what is what are the antibiotics you already consumed? It belongs to which group? So normally antibiotics are classified so many groups. So what are that? So aminoglycosides, okay, penicillins, cephalosporins, tetracycline, then quinolone, sulfonamides, and macrolides. Okay, now we are using which generation of antibiotic? So microbiology, you are a microbiologist, and biotech students may be being here, or biological students. Okay, any type, you know that now we are in 5th generation. Okay, so now 5th generation, okay, now being in 5th generation, so even though now the microbes are treated with once again the old generation antibiotics. Okay, even some microbes are treated with penicillin, cephalosporin, then amoxicillin with claxicillin. Okay, so these are the things mainly isolated from natural sources. Okay, so how? So now we are using herbal medicines in olden days. Okay, just we crush some herbals on the wound, automatically it healed. So after the development of science field, we extract the something from that particular plant. Okay, then we are using the decoction for stomach upset or something. Then we are using the paste for that, the paste created from the plant. Like that we are getting recovery. Am I correct? But nowadays, due to the advancement, we are accurately isolated the particular compound which is specific for healing the wound by various tests. Okay, so by this way only we are isolating the particular compound and standardizing the particular compound, and we are using as a medicine, antibiotic, or a drug. Okay, so this is normally happen. So after a few years later, what happened? So there is no response with that isolated compound. Then the scientists decided and continuously doing the research, and they may attach some other additional groups, esters or some aldehyde groups. Likewise, they are attached that molecules along with the natural compounds. Okay, so natural compounds. There are some other active fractions may be taken out from the other part. Then they may attach with the already known compound like penicillin with the claxicillin, amoxicillin with the claxicillin. So you heard that names. Ampicillin with the claxicillin, Amclax. Okay, or ciprofloxacin with the claxicillin. Okay, then later some of the scientists find these ciprofloxacin, if those who are continuously using the ciprofloxacin, that persons get kidney failure. Okay, those who are continuously consuming drug like cetirizine hydrochloride, those who are, if suppose you may affected with allergic, some of you are allergic in, if you are allowed for long time in AC room, your nose block is happened. Why? Because histamine production, due to histamine production. So you should consume antihistamine like cetirizine or levocetirizine, then desloratadine, then bilastine, like compound you may use it. Am I correct? So among these, any one of, definitely you may use it. Okay? So if you are using the cetirizine, then you get the drowsiness. தூக்கம் வரும் உங்களுக்கு. ஓகேவா? சிட்ரிசன், தென் அதை வேண்டாம் அப்படின்னு சொல்லிட்டு தான் வந்து they neglect that cetirizine, then they are migrating to levocetirizine. It's the living part of cetirizine. Then அதிலும் வந்து பார்த்தீங்கன்னா get tired, and those who are consumed levocetirizine for more than 10 years, definitely they will arise kidney stones. Okay, so then இந்த மாதிரி issues வரனால, so then they migrate into desloratadine. Now currently we are using the antihistamine compound as a desloratadine. So this desloratadine never induce drowsiness. Drowsiness உங்களுக்கு வராது. ஓகேங்களா? There is no tired, no need to take rest. Okay, so automatically it may block the production of histamine. So automatically you are relieved from the black nose, and breathiness, easy breathiness happen. Okay, so this is normally happening. So likewise, so many products are derived from the natural source. Okay, so like herbals, or some marine algae. So marine algae. Now I am concentrating only on marine marine actinomycetes. Why, sir? Yeah. So I mean all microorganisms able to produce secondary metabolites, not only actinomycetes. Why, sir, you may specifically concentrated on actinomycetes. Even Salmonella, Shigella, Vibrio, that organism also produced some secondary metabolites. Okay, so primary metabolites, secondary metabolites you would know. So primary metabolites are metabolites required for their growth, metabolism and their reproduction. Secondary metabolites means it is not required for their life. It may be useful or it may act against others' life for their survival. Okay, so according to this concept, we are procured that secondary metabolites. Then we are testing the various parameters like anti-inflammatory or antihistamine or anti-allergic or anti-inflammatory or anti-hepatoprotective, nephrotoxicity. In any of these mentioned activities, if that that compound persists, means we concentrate that particular compound, we standardize the compound, we concentrate that compound, and we should go through all the pharmacological parameters. Then we use it as a drug, or it may act against others' life, means we may use it as a antibiotic, or we may use it as a antivirals. So this is the phenomena normally happen in the field of pharmacology. Okay, so why, sir, you may select actinomyces? If suppose you may need to extract any particular compound from the marine algae, we should provide the exact environment. Okay, so if suppose you may feel, so which is a sophisticated environment? So your expectation is what? We need to study, no need to write the exam, simply no need to submit the assignments. Am I correct? Really, when I was a student, I am expecting that only. Okay, no need to submit an assignment and simply writing the exam within half an hour. Okay, within half an hour, you need to quit the exam hall, but the result will fruitful, then job will fruitful, and we need a rotating chair, one AC hall, then 2 PA, along with 1 car. It is enough. Am I correct or not? It is enough for you or not? Whether tell, it's boring? It is enough or not? I mean sophisticated salary. So minimum of package around 10 lakhs per annum in starting stage. It is enough or not? Enough? Good. So likewise, organisms also, if we provide the exact environment, appropriate pH, appropriate nutrients. Okay, sir. Why? Because each and every organisms need some unique environment. If suppose you may need to isolate the same bacteria from the terrestrial environment, here the temperature, pH, nutrition availability is somewhat different. If the same organisms you migrate into the estuary, there salted water also as the fresh water is there. Okay, so if that organisms need to survive, what happen? What they will do? It may need some acclimatize their body situation. Am I correct or not? If suppose you may migrate to Ooty, immediately, you may survive easily there. You may survive. Immediately 1 or 2 days, we enjoy. 3rd day, lips are cracked. Am I correct? Skins get dried. Okay? We are not able to do our routine life. Okay? We need gloves and sweaters and other things we need. Then after 15 to 20 days, we acclimatize that environment. Then we could survive and do our routine activities easily. Am I correct or not? Then those who are stayed in woody or some chilled area, that people are migrated into terrestrial area, means there is no possibilities to do their routine life easily. Okay, so they need acclimatization. Likewise, if we may provide the extreme environment to any organisms, that organisms cannot survive easily within a day. So it may survive day by day. It may acclimatize to that environment. Acclimatize na, so change that environment. Namma body adukku thanmaya maathikkiradhu. And the area ku thanda mari maathite, then it may do. So during this acclimatization, automatically organisms produce some metabolites for their survival fitness. That is called primary metabolites. Simultaneously, some metabolites are excreted during their metabolism that is not required for their life. that metabolites may be useful for other organisms or welfare of humans. Okay, so based upon this concept, I am concentrating the marine actinomycetes. That is the first reason. Then second reason, so it is a near to me. So I am in the East Coast region of Tamil Nadu, Chidambaram. Okay, so we could easily collect the samples. Okay, even you may bring the particular, fruits or some materials like ice creams from the bar. So what happened? It's possible to hold the same position when you purchase it after arriving your home. It is not possible to store as like in that host environment. Okay, if we may migrate, definitely it may get somewhat disturbed. It never get their original position. Am I correct or not? So that is the main problem. Okay, that's why I am considering the actinomycetes. So another one reason, so actinomycetes already exhibited in the extreme environment. Extreme environment, okay, sea. Okay, so why? Because sea is the temple for our living. It's a God nature. Why? Because we excreted all the drainage, all the things to the sea only finally. Am I correct or not? So due to the extreme salt concentration, it dissolves all the things and it never give any illness to the human beings, or only it may give the illness to the aquatic environment. Okay, it never give any impact to the neighbors or human beings or animals who are in terrestrial. Okay, why? Because we push all the things to the sea. Am I correct or not? So we may வீட்டில் இருந்து வெஸ்ட் வாட்டர் எங்கே கொடுக்குறோம், கிச்சன் வேஸ்ட் ஆல் தி திங்ஸ் டு பி இன் எங்கே கொடுக்குறோம், நம்ம சீவேஜ் அனுப்புகிறோம். ஸோ சீவேஜ் வந்து நியர்பை ரிவருக்கு அனுப்புகிறோம். ஸோ ரிவர் எஸ்டூரிக்கு போயிட்டு நேராக எங்கே போகுது, நேராக சீக்கு தானே போகுது. ஸோ அப்போ ஆட்டோமேட்டிக்காக அண்ட் ஆல்சோ தி இண்டஸ்ட்ரியல் வேஸ்ட், not only domestic waste, industrial waste also enter into the sea only. So due to the heavy concentration of salt, so it may, in extreme alkaline environment, there is no growth of microorganisms which is illness caused to the human beings. It is the main reason. Okay, so that those organisms able to survive in that extreme environment, that organisms only able to survive and grow well. So in that, actinomycetes is a very known microbe normally present everywhere. In that, marine actinomycetes produce a lot of extreme secondary metabolites, which is useful to us, mainly for pharmaceutical industry as well as cosmetic industry. Okay, streptomycin, you got from where? Streptomyces niger. Okay, actinomycetes. Okay, so that's why I'm concentrated on the actinomycetes, marine actinomycetes, specifically marine actinomycetes. You change it? Okay. So introduction on the pathina. So actinomycetes, marine microorganisms are presently attracting a lot of attention as a novel and prospective source of biologically active compounds. So why? Because so it's already been in the extreme environment. So in salt environment, heavy alkalinity. Okay, so from that environment, suppose you may isolate the organism, it never give any illness to the human beings. That is very much important. If suppose you may need to work in the influenza, if you are not working properly, it may get infected. Okay, but likewise not happen while you are using the actinomycetes. Okay, okay. Marine natural products have been recognized as the most promising source of bioactive substances for drug discovery research. So, why? So already I explained well about why the marine actinomycetes having the potential secondary metabolites. Why? Because it was in extreme environment, so it may, for their survival, it may produce a lot of secondary metabolites. Okay, that metabolites are benefit to us, and as well as so many pigments are produced. Okay, that pigments mainly used for making nail polish and lipsticks and ice cream color making agents. Okay, these all the things are isolated from the marine actinomycetes. Okay, the actinomycetes, actinobacteria are gram-positive bacteria that grow in a variety of environment, have a filamentous shape similar to fungi. Yeah, it's resembling like fungi, but it is belongs to gram-positive bacteria. even they are sized to be 1 to 2 micrometer compared with the bacteria. Okay, but resembling it's in the hype nature is resembling like fungi. That's why it is called as actinobacteria. Okay, next. Actinomycetes distinguish themselves morphologically by forming a layer of hyphae that carry chain of spores. You know that after getting a rainfall or before rainfall, we smell, we are getting a different smell. You may feel that or not. So, right, like the drizzling start அப்படினா, we are getting some different smells. நம்ம மண் ஸ்மெல் வருது, மழை பெய்ய போகுது அப்படின்னு சொல்றோம் இல்லையா? That smells created by that actinomycetes. Okay, especially actinomycetes only reason for that smell. So before getting the heavy rainfall. So actinomycetes have long been known as a major manufacturer of enzyme, antibiotics, amino acids, anti-cancer medications, anti-diabetic pharmaceuticals, anti-obesity treatment, and pharmaceutically and industrially essential compounds. Okay, around 80% of the antibiotics produced by bacteria, including Streptomyces, are produced by this organism. It also capable of producing several active secondary metabolites. So among the antibiotics already discovered, so most of the 80% of the antibiotics are taken out from the marine actinomycetes. Still, we only did 2% of actinomycetes only isolated, only 2%. So remaining is there. Okay, so what is the complications we are facing? So only the environment. Actually, any organisms from their native place, we are migrated to the some other different environment, which it could not survive easily. Okay, that is the problem. So I will tell you later. Next. So Streptomyces is actinomycete is known for producing a number of bioactive secondary metabolites, including anti-tumor agent, antibiotics, then antifungals, antivirals, anti-hypertension drugs, and immunosuppressives. So already I explained well about, so that compounds may be anti-tumor, or anti-hepatoprotective, or anti-histamine, okay. Or it may be used as a perfume industry, or pigments may be, some pigments are isolated from the marine actinomycetes may be useful for ice cream industries and food industries. Okay, so likewise, Streptomyces play a vital role. Only genus Streptomyces has so many lot of species. Normally, thank you, sir. So many things isolated from this Streptomyces. Still now, so many subspecies are isolated to the family of Streptomyces only. Still now, we are isolating the species only. Okay. Then furthermore, marine actinomycetes are presented as a new subject for extensive investigation to find a novel and active natural products which make them a potentially rich and innovative source for new drug development deserving more attention and exploration. So why? So already discussed it. Why we are need to concentrate more attention on the actinomycetes means it has lot of potential active compounds present in there with them. That's why we need to explore that materials means definitely we will be free from germs and healthy life. That's why we are concentrating the marine actinomycetes. Then antibiotics. So antibiotics play a various natural function in microbial interaction in different environment. They can act as weapons or shields protecting bacteria from the competing microbes. So you know that already morning one girl arises the question regarding Helicobacter pylori. Okay, no issues, I will explain. Okay, so suppose you may infected with the Salmonella. Salmonella typhi or typhimurium or paratyphimurium infected with that. As a biological student, what you will do? On initial stage, I mean the first or second day, what you will do? You may get the proper diagnosis, then you may get a treatment, or first of all you may prescribe, I mean you may prefer the OTC product, I mean over-to-counter product. Simply we are getting the medicine from the medical. Okay, I am having fever and headache and vomiting sensation, so you please give. So they are give some pills, I mean tablets. Okay, if cough, they will give the cough syrup, the tablets. So you simply take that, then you continue doing the work. Am I correct? This is good or not? As a biological student, you please don't do like that. Why? Because whenever you may approach any pharmacist in the medical shop, they will give the steroid. A simple, small tablet they will give along with the antibiotic and the cold tablet. You may hear that or you may see that or not. Or simply you may get the injection from the cox, like those who are running the medical shop, they are doing the practice. If suppose you may approach it for any illness, they will give definitely administration of steroid, beta-methasone. They will give beta-methasone. What it will do? So it will totally elevate your total immune system and try to activate the immune system at the maximum level. Only for few days. Okay? Along with that injection, they will give the tablets. Okay? Beta-methasone, betnisol injection. A small ampule they will broken, they will take a 1 cc along with the antibiotic. Definitely you may see or not. எல்லாருமே இன்ஜெக்ஷன் பண்ணிப்போம் அந்த மாதிரி, ஆனால் நம்ம சொல்ல மாட்டோம். சரிங்களா? ஸோ கண்டிப்பாக பண்ணிடுவோம். ஓகே, லோக்கலில் தான் நம்ம இன்ஜெக்ஷன் பண்ணிப்போம், அல்லது டேப்லெட் நம்ம எடுத்துக்குவோம். ஏன்னா போக மாட்டோம், டெய்லினா தான் நம்ம டாக்டர்கிட்ட போகும். ஓகேங்களா? ஸோ ஆட்டோமேட்டிக்லி if you are consuming that type of tablets along with steroid, means what happened? Totally your normal flora will be collapsed. Okay, Helicobacter pylori is a common flora of our stomach. Okay, those who are in ulcer patients யார் இருக்காங்களோ, அல்சர் பேஷண்ட்ஸ் இருக்காங்களோ, அந்த ஹெலிகோபாக்டருடைய பைரோலியோடைய டாமினன்ட் வந்து பார்த்தீங்கன்னா ரைஸ் ஆகிடும். ஸோ பிகாஸ் ஆஃப் இம்ப்ராப்பர் மெடிக்கேஷன், அல்சருக்கான இம்ப்ராப்பர் மெடிக்கேஷன் தோஸ் ஹூ ஆர் கன்சூம்டு, தட் பீப்புள்ஸ் ஹாவ் த இல்னஸ் கிரியேட்டட் பை தி ஹெலிகோபாக்டர் பைரோலி, எ பைரோகிட். ஓகேங்களா? ஸோ வாட் ஹேப்பன்? ஹெலிகோபாக்டர் பைரோலி மே percolate your intestine and make ulcer, and it may lead to sometimes cause as a cancer. கேன்சர் அது கன்வெர்ட் ஆகிறதுக்கு நிறைய சான்ஸ் இருக்கு. தென் அது வந்து பார்த்தீங்கன்னா பெர்குலேட் பண்ணி சிவியர் டேமேஜ் காஸ் பண்ணி ஈவன் உங்கள் இன்டெஸ்டைன் வந்து பார்த்தீங்கன்னா ரீப்ளேஸ் பண்ணுற சுச்சுவேஷனை கொண்டு போகும். ஹெலிகோபாக்டர் பைரோலி வந்து பார்த்தீங்கன்னா உங்கள் இன்டெஸ்டைனையே டோட்டலாக டேமேஜ் பண்ணி கட் பண்ணி இன்டெஸ்டைனை கட்டாகி விழுந்துடும். அந்த அளவுக்கு அது அல்சர் காஸ் பண்ணணும் ஹெலிகோபாக்டர் பைரோலி. ஒன்லி பிகாஸ் ஆஃப் லோ டியூ டு இம்ப்ராப்பர் மெடிக்கேஷன். நீங்கள் வந்து ஆன்டிபயோட்டிக்கை மிஸ்யூஸ் பண்ணுறதுனால தான் இது மெயின் ப்ராப்ளம். ஓகேங்களா? ஸோ இது சிம்பிளாக என்ன பண்ணலாம்? ஹவு கம் வி மே ஓவர் கம் திஸ் ப்ராப்ளம் மீன்ஸ் சிம்பிளாக ஐஸ் பிரியாணி. சிம்பிளாக ஐஸ் பிரியாணி மார்னிங் எடுத்துக்கிட்டிங்கன்னா போதும். தயிர், ஏன்னா லாக்டோபிசில்லி, மீசென்ட்ரிஸ் அண்ட் ஆல் essential bacteria required for to replace the normal flora, all are present in the your curd. ஓகேங்களா? ஸோ இதை எடுத்துக்கிட்டிங்கனாவே ஆட்டோமேட்டிக்லி, ஸோ இட் may dominate the automatically, if its presence is dominant means automatically the Helicobacter pylori number is reduced. It cannot survive there. Intestine வந்து அதனால் சர்வை பண்ண முடியாது. ஆட்டோமேட்டிக்காக recovered from the illness due to ulcer pain. At least it may take 15 to 20 days minimum. Consumption of ice biryani மார்னிங் டைமில் நீங்கள் எப்பயுமே மோர், அதாவது அது வீட்டில் ப்ரிப்பேர் பண்ணணும் தான் இருக்கணும். இட் ஷுட் பி ப்ரிப்பேர்ட் ஃப்ரம் ஹோம் இட் செல்ஃப். ஓகேங்களா? ஸோ வை பிகாஸ் நீங்கள் ஹட்சன் கேர்டு யூஸ் பண்ணுறீங்க, அதில் இருக்கா அப்படின்னு பார்த்தீங்கன்னா மைக்ரோஸ் இருக்கானே இருக்காது. ப்ரோபைட் கேர்டுன்னு இப்போ வருது, அதில் வேணால் மேபி ப்ரெசன்ஸ் ஆஃப் மைக்ரோ ஆர்கானிசம்ஸ் தேர். அது வந்து ஒன்லி கெமிக்கல் ட்ரீட்மெண்ட் தான் பண்ணுறாங்க. ஓகேங்களா? திஸ் இஸ் த பிஸ்னஸ். நீங்கள் if suppose you may due to that pain arises, what you will do? You simply approach a doctor, they will give pyrokit kit. The tablet name is pyrokit kit. If consume that tablet along with the pyrokit, all normal flora will be collapsed. So automatically you may persist with always Helicobacter pylori till your death. You are having the ulcer pain. There is no other go. That is the problem. Okay, so next. So secondary metabolites production by the actinomycetes. Then before that, mismanagement of antibiotic. So before antibiogram test or the exact, at least you should know the exact symptoms appropriate with the particular disease. If suppose typhoid, what type of symptoms normally arises? Knee joint pain, vomiting, yellow layer normally beneath your tongue. That is the main சிம்டம்ஸ். உங்கள் டங்குக்கு அடியில் எல்லோ கலர் லேயர் ஃபார்ம் ஆகிருக்கும். ஓகேங்களா? நீ ஜாயின் பெயின் இருக்கும். ஸோ ஃபீவர் வந்து பார்த்திங்க அப்படின்னா ஃப்ரீக்வெண்ட்டாக ஆஃப்டர் டேப்லெட் எடுத்துக்கிட்டிங்க அப்படின்னா ஒரு பர்டிகுலர் டைமில் இட்மே ஸோ சப்சிக்வெண்ட்டாக வந்துக்கிட்டே இருக்கும் ஃபீவர். ஓகே, ஸோ தட் இஸ் த சிம்டம்ஸ். இஃப் நோ த சிம்டம்ஸ் அப்ராப்ரியேட்லி யூ கேன் கன்சியூம் ofloxacin or chlorompenicol. It's enough for you. And also you should complete the dose minimum of 3 days. If you are consuming definitely 500 mg. Okay, those who are crossed 30, above 30 kg, the persons are prescribed for only 500 mg. If you are consuming 500 mg, you should have that time break of 8 hours. You have that time break of 8 hours, morning and night. இதுக்கு மிடில் வந்து பார்த்தீங்க அப்படின்னா it should be 8 hours. Okay, so along with that you should consume any antioxidant. So you know that antioxidant. So antioxidant are also isolated from the marine actinomycetes. So that antioxidant, if any damage happen in any particular cell or tissue in our stomach, automatically free electrons are formed. That free electrons are migrating to the healthy cells and it need to pair. So it may take the another electrons from the healthy tissue. So automatically damage happen in the healthy tissue. Likewise, the damage happen continuously. So for avoiding that this type of event, you should consume antioxidant. So be aware of as a biological student. Okay. So the secondary metabolites derived from the marine Streptomyces species are unique in that they create novel natural antimicrobials. Most of the antibiotics known to man are produced by Streptomyces species. From the marine source, several potent antibacterial metabolites have been discovered. Streptomyces species produce a wide range of physiologically active secondary metabolites, including the antibiotic. So why? Because anything happen in your body, immediately your physiology system will be collapsed. So you may see a tablet cover. Anybody? No, sir. Simply they are prescribed the medicine. Simply I go through the prescription sheet, whether morning, afternoon or night or before food, after food. Simply I consume or swallow the tablet. Whether it is a pyrogen free or belongs to H drug. Any one of you? No, not aware of that. You please hereafter at least you go through. So what are the things is there? Some tablets are DT tablet. Some tablets are capsule. Why they are prescribing DT? Why they are prescribing capsule? That is very much important. DT means disposable tablet. If you are consuming that type of tablet, it may, before entering the stomach, your colon region, it may diffuse into the tissue. Some tablets have more action than the injections. If you consume, if you are taking the injection, it may work within half an hour. Within 10 minutes, some tablets are working. Some static tablets are working. That type of tablets mostly used as a loading dose. Those who are having the heart illness, heart issue இருக்கிறவங்க loading dose னு ஒரு டேப்லெட் பாக்கெட்ல வச்சிருக்க சொல்லுவாங்க. Aspirin, atorvastatin, then beta blockers tablet. So that tablets mainly prepared in the form of DT, disposable tablet. If that tablet you may just pour it in the tumbler, what happened? Before reaching the bottom region, it totally dissolved. So likewise, that type of tablet before swallowing, just you put it in your beneath the tongue means within 15 seconds it may disperse and it may reach the target region. That type of tablet available. Okay, so all the things should be isolated from the marine actinomycetes mostly. Okay. So the secondary metabolites produced by actinomycetes continue to provide a chemical varied source for the discovery and development of pharmacological drugs as well as the biochemical probes to investigate the mechanisms involved in the human disease. So what we will do, how we isolate the drug next. So then actinomycetes not only useful for the human welfare, so it may also useful for the aquatic animals like crab culture, prawn culture and fish culture. Why? Because the fish culture is the third economic development event normally happening in Tamil Nadu. So in Tamil Nadu coastal regions, prawn culture and fish culture is the most important economic business. Okay, so automatically in prawn culture, some spot diseases are normally happening. Once the spot disease arises, totally the farm will be closed. There is no chance to solve the problem. So totally 15 to 20 lakhs within a day they have lost. Okay, so that problem is also solved by the some of the compounds isolated from the marine actinomycetes. That's why I'm concentrating the aquatic environment also. Okay, so aquaculture is one of the most important economic activities in the world. However, in the intensification of culture practices resulted in the emergence of various diseases caused by microorganisms and non-infectious diseases caused mainly by the deterioration of water quality. Even some of the compounds we are pouring to the welfare of aquatic environment. Okay, but it may not, even though it may not give any good effect to that environment, it may affect the quality of the water. So it may deteriorate the water quality. So what happen? So automatically, if deterioration happen, automatically some other microorganisms or some other living things may be arising in that environment. It may be cause lethal effect to the aquatic environment. That is a problem. Okay, that also minimized by the some of the compounds isolated from the marine actinomycetes. So previously, antibiotics have been used indiscriminately for treating many fish diseases, which resulted in the emergence of antimicrobial resistance. So already discussed. If improper medications, already I said, any antibiotic, if you started, suppose you may started the amoxicillin today morning, you should complete at least 3 days, 6 doses. That is one course. So before that, if suppose you migrated to ciprofloxacin, what happened? That organisms, if susceptible to that amoxicillin means, organisms can get it, reduce their number. Okay, it's not totally killed. Okay, so it may take time for at least 48 to 66 hours. Okay, minimum 3 days. So before that, you migrated to the another antibiotic means what happened? Automatically, that organisms make the plasmids and it may have the resistance against the amoxicillin. Then it may be in silent mode. Okay, then if you are consuming ciprofloxacin, if it is not suitable for that organisms, then the organisms once again reach the infectious dose and cause the same symptoms. Then you may approach a doctor, they will prescribe the amoxicillin, it will not work. The severity of the illness more than the previous stage. Okay, then definitely they will give IV of antibiotics. IV intravenous antibiotics will started. Okay, so why we try to avoid intravenous antibiotics means any antibiotic or any drug you may consume, immediately it may reach the liver. So unnecessary workload to be given to the liver if you are consuming the unnecessary antibiotics or improper medication. That's why I'm advising you. So please, any antibiotics you consumed, you should convey this message at least to the doctor. So according to your information, they will prescribe or they will give the alternate dose. Instead of that amoxicillin, they will prefer the claxicillin or ampicillin or amoxicillin with the claxicillin, combined antibiotic. Because some of you affected with the cold, what you will do? If you are affected with the cold, what type of tablet you are consumed? Cetirizine hydrochloride is a drug. It's an antihistamine. Then amoxicillin. It's right? It is right? Amoxicillin is a right compound for cold. Cold is a viral infection, rhinovirus. Then why are you using the antibiotic? Antibiotic especially donated for bacteria. Okay, so antiviral means antiviral drug, acyclovir, acetylene, these are the antiviral drugs. Instead of that, why we are using the amoxicillin? Any idea? Actually, any viral infection happen, immediately our immune system get failure to react with the virus. Okay, simply interferons may be produced, interferons surround the infected cells, virus infected cells, and it may never allow the virus infected from the virus from the infected cell to the another healthy cell. By this way, automatically, a day passed, automatically the virus get failure. Okay, this is happen normally. So then why, sir, we are using amoxicillin? Even amoxicillin along with the clavinic acid. Amoxicillin with the clavinic acid. We are using clavum tablet. Am I correct? Amoxicillin with the clavinic acid we are using for cold. Why? Why? Because whenever we are arising with a viral disease or viral cold, immediately our immune system get failure to eliminate the virus. But simultaneously, our immune system totally focus on the virus. Automatically, it may get failure to stop the entry of other infections, I mean secondary infections. That's why we are consuming the amoxicillin for preventing the bacterial infections. Okay, bacterial infections. Then why, sir, we are using clavinic acid? Why? Because sometimes we are able to receive the anaerobic organisms. If suppose you are already diabetic or immunocompromised person eats, that anaerobic organism like Clostridium, some of the microorganisms get chance to enter in our body. It may perfect your skins, okay, and make unnecessary problems and unhealing wounds. That's why we are using clavinic acid also for treating the anaerobic microbes. Okay, that's why we are using the combined dose of amoxicillin with the clavinic acid. Simply, if you are find out only the bacterial infections only happen means you could use only amoxicillin enough. Okay, if suppose you are having already some of illness like diabetic or some other illness, I mean disorder you are having, you should consume for the anaerobic tablet also. Okay, this is your choice. Okay. Okay. Next. So antibacterial compound and anti-cancer compounds. The emergence of multi-drug resistant Salmonella species has caused immense public health concern due to the resulting negative impacts. So already I discussed. Salmonella causes foodborne illness which result in significant high death rate. So every year, so more than 5 lakh people died due to illness caused by the Salmonella species. So actinomyces are a group of bacteria species found in soil that create vital biological products, primarily powerful antibiotics. Thus, more intensity should be put into unraveling more potential antibiotics from actinomyces to help reduce the burden of drug resistance. So due to drug resistance, because of our misuse of antibiotics, so we need to search novel products every day. Okay, so there is no other go. That's why we are fetching a new compound from the different sources, not only actinomyces, from the different sources. Some of the people are concentrating marine algae, some of people are concentrating actinomyces, then marine bacteria, marine fungi. So likewise, they are concentrating. Okay, next. Next. All the time up. Next. Yeah. Now I am entering the subject. So how we may produce a compound or develop the compound from the marine actinomyces. How it is possible? So first, sample collection is very much important. Other marine samples, not only soil samples only enough for isolation of proper marine actinomyces. You should collect the some rusted materials in the coastal area. Some rusted materials is there, then normal soil is there, then some soils are touched with water. So you should take the samples in the normal human using area along with the water area. You should collect the both samples. You mix the samples thoroughly. Otherwise, you never get the good actinomyces. Okay. Why? Because if you are get the chance to visit the actinomyces in that, it's like rangoli. Okay, so rangoli மாதிரி உங்களால பண்ண முடியாது. நீங்க என்னென்ன டிஃபரண்டா டயகிராம் வரைஞ்சு வைக்கணும்னு நினைக்கிறீங்களோ அது எல்லாமே பிளேட்ல பண்ண முடியும். Actinomyces. Why? Because they produce pigments, yellow pigments and orange pigments. Okay. So இந்த சாம்பிள் கலெக்ஷன் நார்மலாக வந்து பார்த்தீங்க அப்படின்னா செரனிபெட் மெத்தட் அப்படின்னு ஒரு மெத்தட் இருக்கு. ஸோ நம்ம ஸ்டெரைல் க்ளவுஸ் போட்டுக்கிட்டு வை பிகாஸ் நம்ம நார்மல் ஃப்ளோரா மே இன்ட்ராக்ட் அவர் ரிசல்ட்ஸ். ஸோ செரண்டிபெட் மெத்தட் அப்படின்னு ஒரு மெத்தட் இருக்குது. அந்த மெத்தடில் டாப் சாயிலை எடுத்துகிட்டு ஸோ ஜஸ்ட் ஒரு 10 சென்டிமீட்டருக்கு கீழே இருக்கிற சாயிலை மட்டும்தான் வி ஷுட் கலெக்ட். ஓகே, வி ஷுட் கலெக்ட் தி சாயில்ஸ் ஜஸ்ட் beneath தி upper layer of the earth. Okay, then samples were collected at 10 centimeter depth of the soil surface. Then soil samples were collected at random in a sterile Ziploc cover and stored in refrigerator for further use. இதை வந்து பார்த்திங்க அப்படின்னா எடுத்த உடனே நம்ம லேபுக்கு கொண்டு வந்து நம்ம ரெஃப்ரிஜிரேட் பண்ணுங்கிற மீனிங் இல்லை. திஸ் இஸ் நாட் லைக் தட். ஓகே, யூ ஷுட் பிரிங் தி ஃப்ளாஸ்க். யூ ஷுட் பிரிங் தி ஃப்ளாஸ்க்கு. ஓகேங்களா, ஐ மீன் மூவல் ஃப்ளாஸ்க் இஸ் தேர், நாட் ஏ டீ ஃப்ளாஸ்க். ஓகேங்களா, சாம்பிள் ஃப்ளாஸ்க் இஸ் தேர். ஓகேங்க, ஸ்டெரைல் plastic cover, sterilized plastic cover. So you should collect the sample and immediately you may put it in the flask in controlled atmosphere. Okay, that flask showing the temperature which is normally available in that particular environment. Okay, marine environment and the coastal region la what type of temperature normally behind அப்படிங்கிறது இருக்கு. Then the sample should be collected in seasons, all seasons, not only in cold season. Okay, not only in summer season. So why? Because so due to the environmental conditions, some of the microbes are get died during the summer season, and also the during the rain season வந்து பார்த்தீங்கன்னா due to washing of rain வந்து பார்த்தீங்கன்னா it may leach out. ஓகேங்களா, so you should collect the samples wherever you need to collect the samples. You should collect the samples in different seasons. Then only you could be able to get the exact benefit from the that particular microorganisms. அதுதான் மீன். ஓகேங்களா. Then all the collected samples were subjected to various physiochemical analysis. Why? ஏன் physiochemical analysis பண்ணணும் அப்படின்னா, why we need to do these physiochemical parameters na, each and every environment has some unique characteristics. So pH, nutrient availability, salinity, ஓகேங்களா. ஸோ இது எல்லாமே வேரி ஆகும். அதனால் நீங்கள் ஃப்ளாஸ்கில் ஸ்டோர் பண்ணிவிட்டு சாயில் சாம்பிளை கலெக்ட் பண்ணி அதுக்கு வந்து பார்த்திங்க அப்படின்னா இந்த ஃபிசியாலஜிக்கல் பேராமீட்டர்ஸ் எல்லாத்தையுமே நீங்கள் செக் பண்ணிக்கணும். ஏன் அப்படிங்கிறத சொல்கிறேன். ஓகே, அதை செக் பண்ணிட்டீங்க அப்படின்னா சாயில் சாம்பிள்ஸ் ஆர் ஏர் ட்ரைடு ஃபார் எ வீக் அட் ரூம் டெம்பரேச்சர். ரூம் டெம்பரேச்சர் அப்படின்னா அரவுண்டு பிலோ 40 டிகிரி செல்சியஸ். பிலோ 40 டிகிரி செல்சியஸில் நீங்கள் ரூம் டெம்பரேச்சரில் இந்த மாதிரி இடத்துல ஷேடோ ட்ரை பண்ணிக்கணும். அந்த சாயில் சாம்பிள் பண்ணிவிட்டு தென் கால்சியம் கார்பைடு வச்சு ட்ரீட்மெண்ட் பண்ணிங்க அப்படின்னா இட்ஸ் ஃபார் இன்டியூசிங் த க்ரோத் ஆஃப் ஆக்டோமைசிட்ஸ். அது வந்து பார்த்திங்கன்னா ஆக்டோமைசிட்ஸ் நல்லா வளர்கிறதுக்கு ஹெல்ப் பண்ணும். அண்ட் ஆல்சோ இட் மே ஆக்ட் ஆஸ் ஏ ஆன்டி க்ரோத் ஏஜென்ட் ஆஃப் பாக்டீரியா. அது இதை மெரைன் ஃபங்கையோ இதெல்லாம் வந்து பார்த்திங்கன்னா வளராமல் இருக்கிறதுக்கு தான் இந்த ட்ரீட்மெண்ட் பண்ணுறோம். ஓகேங்களா, ஸோ இது மாதிரி ட்ரீட்மெண்ட் பண்ணிடுறோம். நெக்ஸ்ட், the pre-test samples were used for isolation of marine actinomyces by using starch case in agar with 2 to 10% of NaCl. Why அப்படின்னா NaCl 2 to 10%, 100 ml 2 to 10% பண்ணுறோம். Better you use the marine water. While you are collecting the samples, you along with the can, you should bring the water from the same environment. அந்த environment ஏன்னா அங்கே ppm ரேஷியோ வந்து பார்த்தீங்கன்னா 1000 ரேஷியோ இருக்கும் சால்ட் கான்சன்ட்ரேஷன். அதனால் யூ ஷுட் கலெக்ட் தி சாம்பிள்ஸ் அலாங் வித் தி வாட்டர் சாம்பிள்ஸ். ஐ மீன் அதனுடைய நேட்டிவ் சாம்பிளே எடுத்து வந்து கொடுத்தோம். அதுக்கான ஷெல்டரே வந்து பார்த்தீங்கன்னா மெரைன் வாட்டர். தட் ஒன்லி ப்ரொவைட் தி எசென்ஷியல் நியூட்ரியன்ஸ் டு தி ஆர்கானிசம்ஸ் விச் இஸ் present in the host region. Okay, so you should bring the saline water. Just you measure the starch cation agar in the marine water itself. It is enough. No need to provide the 2 to 10% of normal salt, because sometimes it is not enough to allow the growth of that actinomycetes. So we provide the exact environment, means we provide the marine water for the growth. Okay, then the potential actinomycetes strain were the isolated marine actinomycetes were screened for their antibacterial activity by using the cross-trick method against a bacterial pathogen. Then you isolated the strain from the patients. Along with you should collect the any type culture collections, MTCC or ATCC. Why? Because while publishing the paper in high impact factor, they should arise the questions always. You may compare with the standard strain. Like that they are arising the questions. So don't waste your time. Simultaneously you should collect the samples from the clinical specimen as well as the culture collection centers. Okay, standard collection centers. Then the potential actinomycete strains were inoculated into starch cation broth and incubated at a 20 degree in rotary shaker. Why we are putting it in rotary shaker? In anything, if suppose you need to grow any organisms, you put it in the broth and you may allow to keep it in the shaker. Why? Pardon? Proper growing. Yes. So proper distribution of nutrients happening in the, if you are keeping the culture in the shaker. Okay. If you are keeping the shaker, automatically in broth means automatically it may continuously shaking. So it may provide the nutrients in each and every corner of the flask. So that is the main reason. Why? Because some of the scholars are put the sample simply and kept in the rotary, shaker and switch off the shaker. Simply. Why? Because you said, sir, then I put it there. Like that. There is no result, exact result. We are not able to get it. That is the reason. Then the potential isolates were identified by morphological and biochemical analysis according to the guidelines described in Begg's Manual of Systemic Bacteriology. Simultaneously, if it is active against the our target pathogen means, then we may furtherly do the, once again, we may do the things, cultural characteristics, then biochemical analysis, then 16 is rRNA sequencing. By this way only we could predict the organisms, okay, whether it belongs to which genus. If the organisms sometimes 98% similar to the genus means we are not concluded. 99.9% is only we could conclude that type of genus. Why? Because already we got one culture. We received the result 98.9%, not 99, 98.9. But the publisher rejected my paper. How come? If some microorganism has the ability to similarity 99%, how it is both are unique? Like that they are rejected my paper. Okay, that's why I'm telling. Okay, it's very important. Then next. So then extraction of antimicrobial metabolites by organic solvent extraction method. So you are providing the organic solvent to extract the particular compound. Then crude extract were tested against the bacterial pathogen by well diffusion method. So you are preparing the disc, then you know the antibiogram test. So by this way only you are preparing the disc. Okay. Then mass production and extraction of bioactive compounds, isolation of bioactive compounds by silica gel column chromatography. Pharmacological activity of active fractions, antibacterial and anti-cancer studies. Simultaneously you are using both studies by using animal cell line culture. Okay. Okay. Then purification of active fractions by preparative HPLC, then purified compound were screened for anti-cancer and characterization studies. Characterization of purified compound from HPLC, FTIR and NMR analysis, these all instruments are available in our institution itself. Okay. If anybody doing research, you can approach me, you can do all the things in my institution, okay, Annamal University. Okay. Next. So isolation, just a grammatic representation. So marine sediments, then isolation of bacteria, then simultaneously FESM, then 16S RNA sequencing for the confirmation of the organism's morphological structure, then extraction, identification, then GCMS, FTIR, NMR, then X-ray crystallography studies, we could confirm the compound, specific compound. Okay, then next, then table. Next. So conclusion. Okay, va. So whenever we receive a talk near to lunch, then after the lunch, it's somewhat hard. So no other go. Okay. So myself allotted this session. So no other go. I need to provide all the things, whether you may listen or not. Some of you maybe listen. Okay. So it may helpful for them. Those who are interested to do the research. Okay. The development of new isolation strategies in the recovery of actinomycetes from this environment is of immense importance for ensuring success in this field, and we hope that near in future many novel antibacterial compounds will be isolated for the benefit of human mankind. So இது எல்லாமே ஏன் செய்றோம்? Why we need to do all these things? எதுக்காக படிக்கிறோம்? Why we need to study? For updating our knowledge or for earning money? What purpose we are studying? Earning money or simple sitting after getting a degree, I am wandering here and there, show my certificate like that. For earning money. Okay. So whenever you study any subject or any topic, you should concentrate how to earn the money from the learned subject. That is our motto, students. Motto is, my point of view, if you are studying food, dairy microbiology, while studying curd, how come they are prepared? 100 ml of, 100 ml of curd, minimum of 20 rupees. Per liter curd, how much? You calculate. Okay, but they procure the milk around 32 rupees maximum. Okay, but they are sold out around 120 rupees. Okay, then any product you may add the value, definitely the market ratio will be higher than the previous one. So you study well and please remember this quote, whatever it may be, any topic you should study how to earn the money from this learned subject. Then only it will be remain for your life and you will be entrepreneur if you are studied well or you are concentrated well in particular topic. That is my advice. Okay. So God bless you. Thank you for this opportunity. Thank you all.
So thank you very much, sir, for the insights you had given her for the drug can be isolated from actinomycetes. It's a fruitful presentation and fruitful lecture. So in the lunch break, please, those students who are in poster presentations, you have to display the posters in the lunch break. சரியாப்பா lunch சாப்பிட்டுட்டு அந்த B blockல ஆப்போசிட்லமா 007 ஹாலில் போஸ்டர்ஸ் வந்து யார் யார் கொண்டு வந்திருக்கீங்களோ அவங்க எல்லாம் வந்து பேஸ்ட் பண்ணிவிட்டு யார் வந்து அங்கே லஞ்ச் முடிச்சுட்டு ஜட்ஜஸ் வருவாங்கப்பா எவால்வேட் பண்ணுறதுக்காக. ஸோ அங்கே பேஸ்ட் பண்ணிவிட்டு யார் ப்ரெசென்ட் பண்ணுறீங்களோ அவங்க இது பண்ணிக்கோங்க. ஸோ ஆஃப்டர்நூன் வந்து ஓரல் ப்ரெசன்டேஷன் யாருக்கு இருக்குன்னு பாருங்கள் லிஸ்ட்டில். ஆஃப்டர்நூன் ஓரல் ப்ரெசன்டேஷன் இருக்கக்கூடிய ஸ்டூடெண்ட்ஸ் யூ ப்ளீஸ் மூவ் ஆன் டு தி ஓரல் ப்ரெசன்டேஷன் தி மினி செமினார் ஹால். ரிமைனிங் இருக்கக்கூடிய ஸ்டூடெண்ட்ஸ் எல்லாம் வந்து யூ கேன் ஹாவ் அ லஞ்ச் பிரேக் ஃபார் 1 hour. ஸோ லஞ்ச் ஹேஸ் பீன் சர்வ்ட் அட் ராமானுஜம் தேட்டர். சரியாப்பா அங்கே தான் சர்வ் பண்ணுறோம். நேற்று எங்கே நீங்கள் சாப்பிட்டீங்களோ அங்கே தான். ஸோ அங்கே நீங்கள் சாப்பிட்டுட்டு யூ கேன் கம் பேக் அட் 2:30 at this conference hall. ஓகே, உட்காருங்கப்பா ப்ளீஸ். ஒரு 5 minutes உட்காருங்க. இன்னும் லஞ்ச் வரல, ஒரு 5 minutes உட்காருங்க ப்ளீஸ். ஸ்டூடெண்ட்ஸ் யாரும் பஸ் ஏறி போயிடாதப்பா நம்ம ஸ்டூடெண்ட்ஸ். சரியா 3 நாள் இருந்தால் மட்டும்தான்ப்பா சர்டிஃபிகேட். யாரும் பஸ் ஏறி போயிடாதப்பா. சரியா இன்னைக்கு ஒரு நாள் அவ்வளோதான். இன்னைக்கு மதியானம் செஷன் இஸ் ஆன்லைன். ஸோ நாளைக்கு வந்து ஒரு 3 லெக்சர் இருக்குப்பா. காலையில் ஒரு லெக்சர் இருக்குது, காலையில் ஒரு 2 லெக்சர் இன்னைக்கு மாதிரி தான். அதில் டீ பிரேக் முடிச்சு இன்னொரு லெக்சர் இருக்குது. அதுக்கப்புறம் ஆஃப்டர்நூன் செஷன் வேலிடேட்டரி செஷன் தான். ஸோ வேலிடேட்டரி செஷனில் யூ கேன் ரிசீவ் யுவர் சர்டிஃபிகேட்ஸ். சரிங்களா? ஸோ ப்ளீஸ் யூட்டிலைஸ் திஸ் ஆப்பர்ச்சுனிட்டி ஃபார் யுவர் ப்ரெசன்டேஷன், ஓரல் ப்ரெசன்டேஷன், போஸ்டர் ப்ரெசன்டேஷன். யார் யார் வெறும் அப்ஸ்ட்ராக்ட் மட்டும் கொடுத்துட்டு விட்டுறாதப்பா. இப்போ ஏதாவது ஒன்று போய் ப்ரெசன்ட் பண்ணிட்டு வாங்க. சரியா? சாப்பிட்டுட்டு இங்கே வந்துருங்கப்பா. ஓரல் ப்ரெசன்டேஷன் இருக்கவங்க, போஸ்டர் ப்ரெசன்டேஷன் இருந்தவங்க அந்தந்த ஹாலுக்கு போயிடுங்க. ரிமைனிங் ஸ்டூடெண்ட்ஸ் இங்கே வந்துருங்க. 2:30 வாங்க. போஸ்டர் ப்ரெசன்டேஷன் 2:30லேருந்து ஸ்டார்ட் ஆகுது, 4 o'clock வரைக்கும் இருக்குது. சரியா? So on behalf of our management, faculty members, organizing committee members, I wholeheartedly thank Dr. P.K. Sendilkumar sir for his presence and for his fruitful lecture, for his gracious occasions here. Thank you very much, sir.
ஸ்டூடெண்ட்ஸ் யாரும் பஸ் ஏறி போயிடாதப்பா நம்ம ஸ்டூடெண்ட்ஸ்.
So whenever you study any subject or any topic, you should concentrate how to earn money from the learned subject. That is our motto, students. Motto is, my point of view, if you are studying food, dairy microbiology, while studying curd, how come they are prepared 100 ml of, 100 ml of curd, minimum of 20 rupees. Per liter curd, how much? You calculate. Okay, but they procured the milk around 32 rupees maximum. Okay, but they sold out around 120 rupees. Okay, then any product you may add the value, definitely the market ratio will be higher than the previous one. So you study well, and please remember this quote, whatever it may be, any topic you should study how to earn the money from this learned subject. Then only it will be remain for your life, and you will be entrepreneur if you studied well or you are concentrated well in particular topic. That is my advice. Okay. So God bless you. Thank you for this opportunity. Thank you all.